Okay, welcome everybody, and good morning. This is our fifth and final webinar in the 2020 IFAS Research Forum series. I'm Damian Adams, Interim Associate Dean for Research, and I'm very happy to open this meeting. Today we will be presenting 4 exciting sessions with brief breaks between each. Our first will be Emerging Enterprises, moderated by Bob Hochmuth, Director of the Swanee Valley Agricultural Center. At 9 a.m., Brian Pearson, Assistant Professor in the Environmental Horticulture Department and a faculty member at the Mid-Florida Research and Education Center, will lead a panel on how to successfully integrate research, teaching, and extension, building and maintaining a successful faculty program. At 10 a.m., Kirsten Pell-Stolinsky, Associate Professor in Entomology, who's built a thriving program at the Citrus REC in Lake Alfred, will lead a discussion on managing large and complex research projects. And finally, at 11 a.m., a session on enhancing research through engagement will be led by Martha Monroe, Professor and Associate Director in the School of Forest Resources and Conservation. We have a tight schedule this morning, so I'll briefly remind everyone that you can enter your questions in the Q&A box at the bottom of your screen. Answered questions will appear as they are addressed. Now I'd like to introduce our first moderator, Bob Hochmuth. Bob.
Thank you, Dr. Adams. It's a pleasure to be with you all this morning and moderate our first session on emerging enterprises. I serve as a regional extension agent up at the North Florida Research and Education Center, Swanee Valley, and so this emerging enterprise aspect is something that's been front and center in my extension career, always looking for new alternatives. Last year, about this time, we were planning what became the Emerging Enterprise Summit that was held in early December at the Strawn Center, and we had an amazing participation in that with a number of different faculty throughout the state. In fact, we ended up with 132 attendees at that summit. Dr. Gilbert and Dr. Place had provided an opportunity for Dr. Sam Wisely and I to coordinate that summit, and we were really pleased with the opportunity. I had never met Sam before that and had a real pleasurable experience in organizing that summit. Cassie, if you would maybe put up the infographic now. After the summit, it was obvious that there was a lot of interest in this. And if you haven't seen this infographic, it's available on the Research Dean's website under resources and then under brochures and flyers. And it really is a good visual of showing what happened after that summit. The deans together, along with Dr. Payne, put together over $1 million, in fact, $1.24 million, and were able to fund 19 different teams throughout the state in what became known as the acronym CEDIT. And CEDIT stands for Support for Emerging Enterprise Development and Integration Teams. So it was a very successful summit and has led to some incredible work. There's 175 different investigators in the CEDIT programs. And I'm pleased to say that there's a really strong presentation of extension agents along with researchers. So I think the integration part was well received throughout. What we're going to be doing today in our panel is going to be talking about some success stories. We're going to have 4 panelists. The first 3 are going to be talking, I think, maybe more of a perspective of something that is an emerging enterprise. And then our final presenter will be speaking from more of a perspective of something that was emerging years ago, but now is more of an established and mature type of a program. So I'd like to introduce our first presenter, Dr. Sam Wisely. Sam is a professor in the Wildlife Ecology and Conservation Department, and she's the director of the Cervidae Health Research Initiative. So for us in Extension, that means she's our deer specialist. So Sam, welcome, and it's all yours.
Well, great. Thank you very much. I am the director of the Cervidae Health Research Initiative. And this initiative was started by lobbying by an emerging industry, the deer farming industry in Florida, who were battling some unknowns in a brand new industry in Florida. So Let me start by telling you what deer farming is. So it was really started in about the 1950s or 1960s by Amish farmers in Ohio and Pennsylvania. And the way the industry works is they produce trophy bucks to sell to hunting preserves. And that produces really three streams of revenue. The trophy bucks themselves, which are sold, but also the genetic resources in order to make trophy bucks. So straws of semen are bought and sold just like they are in the cattle industry. And then the hunting opportunities provide outfitting opportunities for people to provide opportunities to hunt. So this is one of the fastest growing rural industries in the United States. Said a study by Texas A&M most recently. In Florida, it is very quickly emerging as a multi-million-dollar industry. There are more than 350 registered farms in Florida, and they're located in 65 of our 67 counties. Most of them are located centrally, or there's a real cluster of them in the central part of Florida. But because this is a newer industry, there's not a particularly good handle on the diseases that affect deer. And when we're talking about deer farming in Florida, we're talking primarily about white-tailed deer. And so while there are diseases that affect native deer or wild free-ranging deer, those diseases tend to have a larger outcome in the farmed deer. And so that was really the impetus for this university industry partnership. In 2012, there was a very large, actually continental-wide emergence of a disease called epizootic hemorrhagic disease virus that killed many, many deer. And there was about $32 million of loss in Florida alone. And that really was the impetus for deer farmers going to the state legislature and asking for help in the form of a university industry partnership. So monies were given to the university in the form of a legislative budget request, and I became the director of what is now known as the Cervidae Health Research Initiative. I view this university industry partnership much like you would the cattlemen's industry with the university, or blueberries in the university, or beekeepers. And so we've really tried to establish a very intimate relationship with the deer farming industry. The first thing we did was send out a survey and really ask deer farmers what it was that they wanted from us in terms of assistance. And it was very clear that three things came out of this. One was they really needed vaccines for some of the largest diseases that affect deer in Florida. And frankly, we didn't even know what the viruses were that were causing these. So even understanding what those were was a very first step for us. Many of those viruses were arthropod-borne, and so developing pesticides, getting them labeled for use on deer farms, and developing integrated pest management strategies has been a very important part of Cherry. And then also providing educational experiences and materials on deer health and nutrition, because many of these deer farmers are brand new. This is not a generational enterprise. These are people coming in naive to an industry and wanting to succeed. So really the meat and potatoes of our program very quickly became what we call the Florida Deer Health Diagnostic Service. So deer farmers can call up a hotline when they have a dead deer. We send out a necropsy team to collect samples. Those specimens are cultured for viruses by our partners, Dr. Lednicki and Dr. Zwalczek, who are our virologists. We have a dedicated wildlife extension veterinarian, Dr. Juan Campos, who assists farmers in understanding these diagnostic reports that are produced and actually makes farm visits to help with animal husbandry issues. And then Dr. Nathan Burkett-Cadena heads up our rapid response vector ID team. So there can be an outbreak of a viral disease, but we have no idea what the arthropod vector is. And so he sends out a team of investigators to trap insects so that we have a better understanding of what the vectors are. The result of this is that we have a very nice rounded program that really touches on the three aspects of the land-grant university. So we have a research program that has produced some really interesting results in understanding what are the diseases that cause death in deer in Florida. This is a phylogenetic tree. All it does is show you the genetic relationships between all of the different orbiviruses. So that's a particular family of viruses that seem to really hit ungulates hard. And so these two diseases, or viruses that cause disease, blue tongue virus and epizootic hemorrhagic disease virus, we now have a library, a cultured library of more than 250 different isolates that's maintained at University of Florida. That has been crucial in helping to develop effective vaccines that are Florida specific for deer farmers. In addition, we have found more than 7 additional viruses that we had no idea impacted deer in Florida. 2 of them were known only from mosquito samples. That's Mobuck virus and Big Cypress virus. And then in addition, we've also found 3 viruses that are brand new to science that seem to also cause hemorrhagic disease in white-tailed deer. Dr. Nathan Burkett-Cadena has also really done a fantastic job in identifying competent vector species, particularly for epizootic hemorrhagic disease virus, which was completely unknown from the southeastern part of the United States. So that's really been a huge feather in his cap and to the Cherry program. And then finally, we've really partnered with, instead of trying to produce ourselves, partnering with vaccine manufacturers by conducting field trials of vaccines that are undergoing USDA licensing. We've also tested some commercial vaccines, autogenous vaccines that were commercially available that didn't seem to be working particularly well. And we have now confirmed that they do not work well. And so we're able to alert farmers when there are things on the market that do not do what they claim to do. We have a large extension output as well. As I mentioned before, Dr. Juan Campos does hundreds of farm consultations every year. We hold workshops, and more recently, we've moved over to webinars. Dr. Jason Blackburn in geography has been working with us, and he's created, as you can see here, a real-time dashboard of disease outbreak in Florida in white-tailed deer farms. So we can tell you where there's a hot spot of EHD or blue tongue virus that's occurring. And then farmers can also see sort of through time, when are the most important times for vaccinating your deer by looking through our case history data. And I should mention that these are all confidential reports, and so everything is consolidated to county-wide data. We have a large social media presence, and we produce lots of printed material as well. On the teaching side, most of that comes in the form of training. Those necropsy technicians I was telling you about are actually pathology internships that we offer. And then in addition to that, we have lots of undergrad, master's, PhD, and postdoc opportunities as well. This has been a really tremendous team effort, not just IFAS-wide, but really university-wide, with multiple colleges involved to really provide a complete picture of what's going on with health and nutrition within the deer farming industry. And so it really could not be done without those folks. Thank you very much.
Thank you, Sam. And Matt, if you'd like to go ahead and get your shared screen going. Just one final comment, Sam, just want to send appreciation again for your amazing leadership and opportunity to work with you in the Emerging Enterprises Summit. It was great to work with you. The next presenter will be Dr. Matt DiMaggio, and Matt is an assistant professor at the Florida Tropical Aquaculture Lab in Ruskin. and that is part of the School of Forest Resources and Conservation. And an extension EAS, that means that he's an ornamental fish specialist. So Matt, take it away.
Thank you, Bob. So what I'd like to talk to you today about is the expanding role of marine ornamental aquaculture and the industry within Florida. Let's see if that advances. Okay, sorry. So Florida has a very diverse aquaculture industry comprised of numerous commodity groups. This includes things like ornamental fish, which I'll be speaking about to you today. Things like mollusks, clams, and oysters are a common one grown here in Florida, as well as things like reptiles, alligator farms, there are turtle farms in the state. Also, things like shrimp, crayfish, aquatic plants for water gardens, and we do have some food fish in the state. There are about 1,000 licensed farms as of about 2018 by the Florida Department of Agriculture and Consumer Services, but we'll be focusing today on the ornamental fish industry. So that's fish that you would buy, for example, in a pet store. So Florida ornamentals represents the largest segment of aquaculture within the state by value, a little under $29 million as of 2018. And we have about a little over 100 producers in the state. Now, by and large, this industry is dominated by freshwater ornamental production, freshwater ornamental fish. So in that picture there on the right, you'd see some of those fish that you may see in a pet store, things like tetras or angelfish or algae eaters or plecos, that big fish that you see there in the center. We grow about 400 or so different varieties in the state of freshwater species, but marine species, for example, like Nemo, the clownfish, you may be familiar with there on the bottom left hand, they're sort of an up-and-comer. There's about anywhere from 75 to 100 different species in culture at one time within the state, but this does represent a growing market and thus an opportunity for farmers within the state to expand their operations. So why do we want to look at marine ornamental fish? Well, first, the supply is predominantly wild. Current fish that are in the trade, current marine ornamentals, are generally harvested from wild coral reefs around the world. And the diversity is actually quite staggering. We bring in about 1,800 different species of fish every year into the United States. And of those 1,800 species, that represents 24 million individual organisms that are imported. So accordingly, there has been some concern regarding the sustainability of these practices, especially some collection practices. In the picture on the top right there, you see a ornamental collector who's actually using cyanide to stun fish on the reef to make them easier to capture, and that's not necessarily the most sustainable of collection practices. And then also conservation. Is there a role that aquaculture can play in promoting conservation? Can we remove pressure off those wild stocks by culturing that product? The markets too are quite large. There's 700,000 US households within the United States that maintain marine aquaria, and we've actually seen an uptick in sales and in that interest during the pandemic. People have actually started to keep marine aquariums, it seems, more. And there's over 2 million home and public marine aquariums globally. And finally, we're looking to diversify aquaculture in the state by developing another commodity group for our farmers, making them more resilient in the marketplace. So why is growing these fish so difficult? So if we were to compare this to common animals that are produced, for example, like pigs, when pigs give birth, the babies somewhat resemble the parents. The parents can provide nutrition. Chickens will lay the eggs hatch out, and we have a baby chick that can go right to a commercialized, readily available diet. Well, when I work with blue tangs, for example, they spawn their eggs, the eggs hatch out, and we are left with usually a 2 millimeter or less than 2 millimeter organism that is essentially a yolk sac and a primitive vertebral column that is incredibly fragile. So we have a lot of obstacles to overcome in raising these organisms. And those primarily deal with reproduction, getting these fish to reproduce on a consistent and reliable basis. And then also finding efficient ways to produce those larvae to promote survival and growth. So some of those bottlenecks that we run into, that we want to identify for farmers as we sort of put together these cookbook recipes, these protocols on how to raise all these different species, are things like, for example, first feeding. When the larvae transition from relying on that yolk to exogenous feeds to actually going out and hunting small zooplankton, like you see there in the top right-hand picture, that's a time when we would see major mortality. The fish have to be able to actually go out, capture that food particle, be able to physically ingest it. It has to be of the appropriate size for them to fit it in their mouths. And then they have to be able to make use of that organism. They have to be able to assimilate that nutrition. Another time period where there can be mass mortality is swim bladder inflation. That's the organ that actually helps with buoyancy in the fish. In that center picture, that circular clear spot there in that larvae. That's another opportunity for mortality. And then finally, metamorphosis, where the fish transitions from that larval state in the second picture to more of a juvenile adult state in that third picture on the bottom there. In this case, this is a millet seed butterfly fish. Another opportunity again for mortality to occur. So we want to elucidate when these developmental periods occur to try and ameliorate that. So the easy ones have been done, so to speak. Things like Nemo, the clownfish, a very easy fish to grow, which is great. And things like damsels and dottybacks, seahorses, for example. A lot of these fish are already in culture at commercial facilities around the state. And the common thread between all of these groups of fishes is that they have large eggs, which in turn produce large larvae. There's some degree of parental care, and they consume a zooplankton called the rotifer, which is cheap and easy to produce. In contrast, what we're working on at the Tropical Aquaculture Lab is research focusing on the difficult fish, things like the Pacific blue tang that you see in the top right, or Halichories wrasses, the Melanurus wrasse, which you see on the bottom right-hand corner. These have strong market demand, but no current aquaculture protocols. And the common thread with these fish is that they all produce very small eggs, which in turn produce small, fragile larvae with no parental care. And these fish commonly rely on a zooplankton called a copepod, which is difficult and expensive to produce. So just to go through real fast, those two species we're working with, the Melanurus wrasse, it's very popular in the aquarium trade. It currently retails for about $30 to $100 a piece. And if you equate that to a per pound basis, you're looking at about $450 a pound. So pretty impressive if you put that on sort of a food fish scale. It's one of the top 20 most imported wrasses into the US every year. And again, we're trying to define those protocols for spawning and larval rearing. We've been pretty successful at developing preliminary production protocols for this species, primarily looking at feeding, what do they eat, when do they eat it, how much food do we offer to them, and then basic ontogeny. For example, you see that figure on the right, where they go from 1 day post-hatch at the top, again, that primitive vertebral column, all the way down through 45 days post-hatch, where they resemble their juvenile form and are close to what you'd see in a pet store. We've been relatively successful in beginning domestication with these species with both an F1 and an F2 generation, and we're starting to transfer some of this information to industry so that they can get started with trying to raise this species. The Pacific blue tang, the 12th most imported species overall into the United States, they retail for about $65, and again, very high on a per pound basis, $590 a pound if you equate it that way. Markets are heavily traded. This was the fish that was featured in the Finding Nemo movie and the subsequent Finding Dory movie. There are questions concerning conservation and collection practices with this fish. And again, we're trying to elucidate those production protocols. What I want to just go through here real fast is just some of the development that we've been able to document and describe with this fish, starting out with 5 days post-hatch and to 15 days post-hatch, where you see the beginning of some of those spines that the fish is known for. By 19 and 27 days post-hatch, those spines start to elongate, the fins start to form, the body starts to deepen. These are all critical developmental time points that we need to know when they happen. Again, increasing body complexity, fin formation as those fish start to sort of grow into their spines. And then from day 46 and day 55, 54 or so, they start to take on that characteristic blue pigment that you expect of the species, starting to end that larval period. So we were the first ones to successfully culture this fish from egg to juvenile in the world at the University of Florida at the Tropical Aquaculture Lab. That first run, we were able to raise 27 out of 50,000 eggs for a 0.05% survival rate. Not very encouraging, but for us, we took it as a success, as this hadn't been done before, and this was proof of concept that the life cycle could be completed in a laboratory setting. So we were very pleased, and we've done it subsequent times since then, and we're trying to refine that process. It was great that while we were doing this research, there was also a movie that grossed $1 billion with the same feature character, which actually helped to get that research information out to the general public, which is another stakeholder of ours. We want to educate the public in terms of where their marine fish come from, start that discussion so they ask, where their fish are sourced from, are there sustainable options, what's going on with aquaculture. We've been able to get that out through print, radio, TV. And one of the other things we're really proud of is our partnership with SeaWorld and Busch Gardens, where we have videos up describing this process at UF, the rearing process at in Orlando, San Antonio, and San Diego. And these videos have been viewed by over 20 million visitors since 2016. So it's a really great venue to get that information out to the general public. We've also been pretty successful at domesticating this fish as well. We have F1 populations that are spawning that we're running research on, research experiments right now to try and optimize some of those production parameters. And again, we're working closely with the industry, the Florida Tropical Fish Farms Association, to try and transfer these findings to producers to get this production off the ground. So to conclude, success for us really lies in commercialization of these species. We have proof of concept, which is great. We're trying to get this technology into the hands of stakeholders as quickly as possible. But we also want to make sure that we're able to educate the public and communicate our science effectively to promote aquaculture as a conservation tool as well as a benefit to farmers within the state. We're excited about the growth and the continued research with marine ornamentals in the state. And we think that Florida is uniquely positioned to continue on as a leader of marine ornamental production, both in the United States and globally. And finally, we just want to make sure that there's always a sustainable alternative to wild capture through aquaculture. This work was supported by a whole host of people, which it could never have happened without their support. And if we have any questions at the end of the session, I believe Bob will take some. Thank you.
Thank you, Matt. Very, very interesting. Brian, if you want to go ahead and get your share screen going, I'll introduce you. The next speaker will be Dr. Brian Pearson, and Brian is an assistant professor at the Mid-Florida Research and Education Center and a faculty member of the Environmental Horticulture Department, and is in charge of the Medicinal Horticultural Lab there at Apopka. So I could say that he's the Medicinal Horticultural Specialist, but for the next 10 minutes or so, he's going to be the beer specialist. So Brian, take it away.
Thank you very much, Bob. And I appreciate the opportunity to speak here today to share with you a journey of an emerging enterprise that I didn't realize I was embarking on when it started. And so the presentation today is really focusing on my experience, and I really wanted to focus on the life cycle of emerging enterprises. So in my opinion and my experience, I could really sum it up with three key components, I think, that make a successful emerging agricultural enterprise. And I'll speak about all of these in greater detail and relate it back to the HOPS project and then talk about some milestones. But the three key points would be passion and possibility. The product should have market potential and have good collaboration and support. And so what do I mean by passion and possibility? I think it's important and critical to collaborate and identify possible research objectives, both with your colleagues and with our stakeholders in the state. And whenever possible, I think it really helps if you can tap into a passion that exists, particularly in our stakeholders. And so you'll see here, I use the term inner nerd, and this is where we get to kind of nerd out and share our passion for what we do with our stakeholders. And I think if you can accomplish that, one, it helps to identify and refine your research objectives. Plus, it helps to get that industry buy-in and works wonderful for extension opportunities as well as for teaching opportunities. I think any emerging agricultural enterprise should also have some focus on market potential. The new enterprise should be realistic because ultimately we're going to ask producers to assume some risk in taking on these crops, in the case of hops, producing hops for the state. If possible, Matthew just mentioned a proof of concept is very helpful and critical, and you can use this also to communicate with stakeholders and to do it often. Lastly, none of it's possible without collaboration and support. So I think it's really critical to form a strong team, have interdisciplinary projects where possible, And if you're successful, I would warn you to be prepared to be overwhelmed, which is a good problem to have. So let me go ahead and talk about each of these a little bit more in depth, and particularly how it relates back to the hops research. So passion and possibility. This is where it's a matter of getting involved with your stakeholders. Ultimately, most of us as researchers probably agree that there's endless research possibilities. However, new enterprises are going to be more likely to be successful if we have stakeholders and can connect with them that are passionate, as passionate as we are about what we do. So this is where sharing that inner nerd passion comes back into play. And so back in 2012, I shared my view that the local hops I got were stale and kind of not so impressive. I thought, why not? I'm a horticulturist. I should be able to grow them. And so I met up with cold calls, several brewers. In the case here, you'll see the folks at First Magnitude. Met with them and shared that nerd passion with them, and they agreed and helped refine sort of my research objective is, could we produce hops? How much do you need? And in what forms and qualities are you looking for? And so that was a really good marriage between them as stakeholders and me as a researcher, with the emphasis being on local ingredients. Sort of validated and again refined the work that I had proposed to do. Switching gears, market potential. I think any new enterprise should be somewhat realistic. And if you could ask the question, can the new enterprise be adapted into current production systems with relatively minimal amount of new equipment and procedures, I think it has a greater chance of success. In the case of hops, most farmers have most of the equipment necessary to produce and cultivate hops with very few and minor things that they needed. And that became, again, part of our research and our extension focus for how to educate our growers and to help them overcome those barriers. Ultimately, again, to be successful producers are going to have to be willing to assume some level of cost and risk. So it's critical that we work with them to help identify those and make those part of our programmatic goals. Emerging enterprise research should aim to develop both new knowledge and, as Matthew said, a proof of concept. And so this works excellent. One, because again, it refines what our research is. As we're building this proof of concept, we find issues, and those become our research objectives to try to solve them, as well as an excellent opportunity for extension. And so at Mid-Florida Research Education Center, we have a hop yard. We've had in-service training events for extension agents, as well as open field days for the general public. And this helps really to develop industry support, both for political support, letters of support for grants, as well as direct financial donations. And we've been lucky enough to receive both. The third tier, I think, in my opinion, looking at emerging enterprise projects, what makes them successful, is a level of collaboration and support. Anytime you have an interdisciplinary project, I think you have the best chance for success. And speaking personally, it makes it a lot more fun because you end up having a lot more expertise and it adds a richness to the work that you're doing. I do get asked several times, well, how do you end up forming this group and working with individuals? This is, again, where I would say insert that inner nerd passion. I think each of us are probably guilty of going to professional associations and kind of nerding out where we talk about what we love. And so cold calls to individuals in other departments like Charlie Sims, Food Science and Human Nutrition, led to a collaborative opportunity, which I'll speak about next. With agricultural commodities, and especially with an IFAS, we've got an amazing diverse array of different disciplines and expertise. So I think we're lucky in the fact that we can tap into this. Lastly, forming a team, when it's organic and authentic, it really seems to work well. And team building 101, personality, openness, and making sure that you create a safe environment for the team, I think is important as you're throwing ideas out and being able to have other colleagues that can critique your work and vice versa to refine it and make it better. There is a lot of momentum, I think, required, especially to kick off and start an emerging enterprise. So having a team kind of helps with that momentum. And I said previously, if you're successful, prepare to be overwhelmed. This is something certainly with hops, the amount of interest we received was very overwhelming to me, and we've seen that recently with hemp as well, which is a good problem, but it certainly helps when we have a large team to help field this. So as promised, I said I'd talk a little bit about the hops project, from how it started, where it currently is, and where it's going, to kind of talk about that life cycle, a little bit more about projects. So I said previously, this was sort of a crazy idea that was hatched up on a Sunday afternoon, brewing beer in my backyard. And on Monday morning, I decided I was going to start growing some hops at the Mid-Florida Research and Education Center. So the picture on the left shows what this looked like. And as crummy as this looks, I'm still incredibly proud of this. This is assembled with 3 pieces of irrigation piping that were supposed to be thrown out. I recycled and reused them. purchased rhizomes, some pulleys, string, and fertilizer with my checking account, and I went to town to see if I could actually grow them. And again, that's that proof of concept. Could it be done? And along the way, collecting data. And lo and behold, despite a lot of people's belief that it couldn't be done, we were able to cultivate hops. And the interesting point was how different the different varieties ended up performing. And so that, again, gave us that initial information. So between 2012 and 2015, I kept investing in buying more plants and more to the point that I was probably pressing my marriage. And so by 2015, it was great that the Florida Experiment Station made some funds available so that we could actually build a large tall trellis system here at the Mid-Florida Research and Education Center. In 2016, now at this point in time, we'd already amassed enough information, publications, and had that critical mass to go after grants. So collaborating with 2 faculty members in IFAS, we were able to secure a grant for 2 years, beginning in 2016, to lead a large varietal trial. That allowed us to develop, again, enough hops to take it to the next step, another proof of concept. So it's that life cycle repeating itself. Another cold call, 2 of them, in fact, one to first magnitude, saying that I've got a lot of hops, could we brew a beer with it and maybe do a extension or outreach event with it? And 2, going back to FDACS, who had funded the work and say, hey, we're at a point now that our industry is beginning to farm hops. Could we work with you, or would FDACS entertain a fresh from Florida, made with Florida hops label? So in 2017, we were able to get this label working with FDACS, working with First Magnitude Brewing. We released the beer here, the Apopka Hop Pale Ale, and the excitement around it just blew my mind. They had brewed about 30 gallons, and it sold out in just over 20 minutes. So to me, that was really a success. And thinking from 2012 to 2017, that was one life cycle that we were able to accomplish and to show return on investment. I still had a lot of hops over, even after the Apopka Pale Ale event. So as I mentioned earlier, a cold call to Charlie Sims, working with Charlie and 2 other talented faculty at IFAS. We ended up on a committee working with Sean Krieger, who did volatile assessments and panel sensory assessments of our hops. And this goes back to the first part of local ingredients. And so with his work, we were able to document the fact that Florida grown hops have unique flavors, or in this case, actually aromas, that speak to that local interest that both I had as a beer nerd as well as our farmers. Fast forward to 2019, working with Alfred Waugh, new hire at Mid-Florida Research Education Center. He's spearheading a hop breeding effort to hopefully overcome the mismatch between the environment and the plant. And so this is exciting new work, and I'm hoping that this eventually becomes sort of similar to blueberries, where we could have varieties that are specifically cultivated for Florida. So again, it makes it easier for producers not to have to worry about lights or manipulating the environment. And so this is a 2-year grant. And then lastly, 2020, this is a story that actually broke last Friday to last week. A young gentleman, Sebastian from St. John's Hops, hop farmer in the area. But what I found interesting about this article is not only is he farming these hops, proof of concept, but he's also a veteran dealing with PTSD and has opened up his hop yard for horticultural therapy, which I thought was just a whole other, again, life cycle that could be a jumping point for additional research. So with that, where are we going? UF IFAS Brewing, Distilling, and Fermentation, 2020 and beyond. We've really formed a core group, and also been some new hires that I think all kind of capture that passion and possibility, the market potential, and collaboration and support. Some of these individuals, like Alfred, will be handling some of the breeding efforts with hops. Recently, we've had hires like Andrew McIntosh and Catherine Thompson-Widrick in food science and nutrition, who are going into brewing and distilling. And as well as I mentioned, Lee Deal, and possibly working into horticultural therapy to see if we can bring a new dimension to this effort. So with that, I'm going to say thank you, but since we're talking about beer, I think it's appropriate to say cheers. I would like to recognize the financial supporters that made this work possible: the Florida Ag Experiment Station, FDACS, and First Magnitude Brewing Company. So with that, thank you, Bob.
Thank you, Brian. Very interesting presentation as well. Patricio, if you want to go ahead and get your share screen going, just a thought there at the end of the presentation that Brian was having in evaluating these different beer products through the Seeded program. I'm familiar with a couple other aspects that also have a beer theme, and there's going to be some work done. Lincoln Zotarelli and Charles Barrett have a project here on malting barley, and also Jeffrey Meru has a Calabasa program in his breeding repertoire. We have a project here that we're collecting and sending Calabasa down to campus next week for a pumpkin type beer test as well. So a couple other interesting sidelines to what Brian is doing with hops. Just as a reminder for everyone, if you have questions as we finish our final presenter here, we'll take questions afterwards, and those questions should be entered into the question and answer section, not the chat area. So if you have questions for any of our panelists, go ahead and prepare them as we go through this final speaker. Our final speaker will be Dr. Patricio Munoz, and he is an assistant professor in the Horticultural Sciences Department and a blueberry breeder, is in charge of the blueberry breeding and genomics lab. And just want to let you know, Patricio, that I'm in the fan club of blueberries. My wife and I every year pick somewhere between 150 and 200 pounds from a local U-pick operation and stock one of our freezers. So looking forward to having your presentation here.
Well, thanks for inviting me to talk. You know, my title basically is the summary of the whole talk. Given that we don't have much time, you know, is the outline of the whole talk. And what you were just saying, and what Brian was mentioning, and the other speakers before, you know, the passion that the fruit or the crop that you're working with, or the species that you're working with, make this moving. I'm being fortunate to work in 3 different crops, you know, forest trees, in the part of the consolidation over the rise of the industry, also in forages, where I participated in the starting of the industry for a couple of crops that didn't exist in the state, and in the case of blueberry, of a very consolidated industry that has a different type of challenge that I'm going to be talking about a little bit. So to talk about blueberries, first I need to present the species that we're going to be working with or talking about today. But importantly, you know, I'm going to give you a few facts that probably are going to be very relevant for you, but, you know, you're going to have to learn them. So there is 3 different species actually that are sold in the supermarket as blueberries, but in reality, there are different species. So you have just the common names here: low bush, rabidice, and high bush. In the high bush, we have 2 different categories. We have the northern high bush and we have the southern high bush. Well, the southern high bush are fairly new. The northern high bush are the ones, you know, that they are commercialized globally, or the high bush in general commercialized globally, and the ones that have better characteristics in the past. Another fact that you need to know before we go more into the content of the talk is that in Florida we have several species of wild Vaccinium. So, for example, here we have Vaccinium deroy over green blueberry, Vaccinium eliotii arboreum that actually looks like a tree, and Vaccinium staminum. Now you will say, why are these species important? They're very important and relevant actually for the breeding program, and they were used significantly in the past. But for example, just to give you an idea how important they are, you know, if you blend fresh blueberries, the ones that you buy in the supermarket, you're gonna get this white juice as you see, you know, in your left of your screen. But actually, when you use some of the other blueberries that we have native to Florida, for example, the Vaccinium staminum or hybrids of Vaccinium staminum with high bush, blending fresh blueberries, you can get, you know, this purple or reddish color. Now, another fact that might be relevant, but probably you're gonna learn, and maybe it's gonna be useful for you. Whenever actually you buy those fresh blueberries in the supermarket, and if you freeze them and then you blend them, actually you can obtain the red or purple smoothie that you are looking for. Well, the story goes like this. Northern high bush could not be grown in Florida because of the different climate conditions, and then actually they could not achieve the chilling hours requirements. So that means the amount of winter that they need, or the amount of cold weather that they need to basically blossom and produce fruit. So that was not possible in the past. And by just putting all this together, like I was telling you before, At the University of Florida, the role of IFAS was that they put together, northern highbush, the species I was telling you before that, it has more interesting fruit characteristics, qualities, and etc., with wild Florida blueberry species to create what we know today as the southern highbush. So it was born here in Florida. Now, what was the role of IFAS in all this? Well, the domestication of these species in the north is not very far, actually. It was just happened at the beginning of the 19,000s. And then, the process of domestication, since they select the first plants in the field, in the wild, actually, and then they learn how to grow them, it was around, 30 to 40 years, more or less. So the emergency, we can say the emergency on the industry, in the case of Florida, it happened between the 50s and the 70s, more or less. What happened in this period? Two visionaries, breeders of the University of Florida working in horticultural science in IFAS, collaborate with George Darrow from the north in the USDA, actually to study and to hybridize these two species, the one from the north and the natives from Florida. They have many failed attempts with different species, and also, other participants in Georgia and Mississippi were helping in this process. But it was, a very fortuitous and a very, a serendipitous kind of casualty. They collected two clones that they thought there was one species, but it turned out to be, it was a different one. And they collected in Winter Heaven. And then Ralph Sharps made the crosses with this species, was Vaccinium derowii, basically. And one single clone, one single plant actually that has a tendency to fail in the genetic composition of the gametes. And actually because of that, the crop was born. What happened, technicality, what happened is that this plant failed to produce the gametes as it should. So it was a diploid plant, and it should produce a gamete of one single haploid of one end. And then what happened is that it failed to produce the gamete, and then it produced two gametes with two chromosomes, and then it will match the species of the gametes with from the north, and that created the species that we know today, or we crop. So the second part, so the creation, it happened, and then, around the 70s, the first crop, and then the rise of the industry happened when Ralph and Wayne actually produced the first cultivars in 1976. And from that moment, there was a point that they were able to produce a lot of plants. They were able to generate populations that were significant for a breeding program. However, they didn't have the capacity of the people or the resources to move forward with all this research. And in that point, the collaboration was key. The collaboration with a couple of producers in the States, where actually they did all the production, they did all the maintenance, and they all the in-kind funding for putting this plant breeding program together. So this is a second part where, the collaboration and the symbiosis that you can say, between the University of Florida researchers with the farmers, producers, and stakeholders, helped to create this vision. And obviously these were visionaries, the same as the researchers here. They had passion at the same time. The growers was the same for them. After that, there was kind of a explosion on research, and then talking about the benefits of antioxidants and anthocyanins in particular. In this case, we can show that blueberries have the highest levels of anthocyanin. And then because of that, global production started, more or less driven by this, desire to eat, healthy fruits. And then not minor, we eat around, this is a statistic of 2016, we were eating around 68% of the global production of blueberries here in the US. The consolidation of the industry, we can say that happens between 1919 and 2012, when actually what was the role of the University of Florida breeding program in this case is that keep releasing cultivars that could, help the producers achieve what they wanted to achieve in terms of quality, in this graph, we can see every single blueberry cultivar release that is being published up to 2017. And for example, the firmness of the fruit. And the ones in blue are mostly Southern highbush, the ones that we work with. And most of the blue actually belongs to the University of Florida blueberry breeding program. So right now, around 70 to 76 more or less of the global cultivars that exist, they track back to our pedigree. The Florida industry right now is around 50 to 60 farms, around 5,400 acres. How do we know that? Because we visit each of them after every season. And around 28 to 24 million pounds of fruit are produced, and we are the first in the state. So we actually, because we are the first producing in the state of fresh fruit, we have a special price that's better than in other states. In terms of the research, this has helped us to create agreements, and then actually our genetics is used in many different, in almost every continent, but Antarctica, we say, in many different countries, and then we collaborate, and then in some of them actually we work together to generate new cultivars specifically for those places. Now, after the consolidation of this industry, obviously comes the challenge. The challenge because everybody's producing, everybody's sending fruit to the US because it's the largest consumer of fruit globally. And then we need new research, we need new methodologies of growing, for example, new systems of production. This system is not new for other crops, but actually it's new for blueberry. And here we were able to achieve with these containerized systems in tunnels, we were able to achieve 5 times the production levels that you can achieve in traditional systems. We keep working with wild blueberries because they still have a bunch of new traits that we can bring to the cultivated crop, and then obviously it's going to benefit the producers and also the consumer. We keep working in methodologies or developing cultivars to decrease the cost of production for producers so they can compete in nowadays very challenging and global market. And the other thing is that we need to develop new ways for them to differentiate. And then one of the things that we've been working a lot is being the understanding and the capacity to predict the flavor of new cultivars that we're generating. And I think finally this year we basically achieved one of these goals. With all that, I will say stay healthy, Blue, and then I'll be happy to take questions afterwards. Thank you.
Thank you, Patricio. We are going to be a little bit short on time. There's a lot of information that's been jammed into this period. I think I see 2 questions that are in the question and answer session. We'll try to take those and maybe be completed by 8:55. The first one is from Dr. Dittmer, who is asking about if any one of the panelists are familiar with industries or crops that have failed, and or even other enterprises, and why they may have failed. Would any of the panelists like to take that one?
I could talk very briefly about it. Whenever I was working in forages before, we tried many different species, and some of them, they didn't make it all the way through. So out of probably the 4 or 5 species that I tried to introduce into the state, I think 2 of them are the ones that continue to move forward. Why? Because they are just not adapted, which is the case of the blueberry. So that will take, investment and then time to make it happen.
Thank you. And I think my perspective, those challenges have been a lot of folks that get into thinking that they may be able to grow something that just really is not adapted to the area. And then it requires research to figure out if there's a way of modifying the environment or the climate and putting it into some kind of an protected ag type of a situation. So those would concur, I think, with what Patricio is saying. The other question also is for Patricio in relation to blueberries, and the question about the premium that we receive here in the United States, but what about the rest of the world? Does Florida blueberry harvest directly compete with other parts of the world like Peru?
In terms of, I mean, you can compare in different ways. And actually, this research that we are doing regarding containerized production it was born out of my frustration visiting all these places. The background photo that I have there is actually 2,000 hectares of production in Peru. So what happened is that I go to all these different places, and the same genetic that we develop here, they produces 4 to 5 times more than we produce here in Florida. So they are using different production systems, so they can have more volume than us. The production system is a little bit different. In regarding quality, we are ahead so far, but these are industries that are huge in these countries with huge investments. And then they are moving very fast. And I think now every single, for example, plant breeding program is in Peru and in Mexico, and then trying to make better cultivars with better quality to compete with what we do here.
Thank you. Just have one quick question to Sam. If you could maybe address this. This seems like a really unusual situation where the survey industry really didn't exist before, and just be interested in how you find those people. How have you communicated with them? Have you organized your extension educational program with them? If you could briefly answer that one, and then we'll close her down.
Sure. Well, we're very lucky in that Deer Farmers have to be registered, and that is a public document. And so we really surveyed the entire industry and just let them know that we were there. And then they have organized loosely. And so there are 2 Florida organizations for farmed cervids. And so we really hit all of their public meetings and professional society meetings, really telling them about our service. It took a lot of trust. We had to do a lot of trust building in order to get them to actually send us deer samples. There are lots of diseases that can actually have people's herds depopulated. And so it's been about 5 years to get really them on board. And now they are very, very supportive of what we're doing.
Thank you, Sam. Just a final conclusion, Dr. Adams, thanks for having me today to moderate this, and just kudos to Dr. Gilbert for being the brainchild of this whole emerging enterprise program over a year ago. So thanks for allowing an extension agent to be moderator in your research forum. Thank you very much.
All right, thank you very much, Bob, and thank you to everybody on the panel. I did note that there is one question remaining that's in the Q&A, and that could perhaps be answered by text later. So I just wanted to point the panel in that direction. So if everybody could just sit tight, we're going to take a few minute break and then come back at 9 o'clock for our next session. Thank you.
Everyone, for your attendance today. My name is Brian Pearson, Assistant Professor in the Department of Environmental Horticulture, and this will be the second session of the IFAS Research Forum today on how to successfully integrate research, teaching, and extension, building and maintaining a successful faculty program. Today I'm co-moderated and joined by Dr. Ann Matthews, an Associate Professor in the Food Science and Human Nutrition Department. Her and I have put the session together to really focus on building better links and connections within your program to maximize impact and success. We've identified 3 speakers today who have done an exceptional job at developing complementary elements and their associated outputs within their faculty programs. For today's session, please hold your questions until the end. Hopefully we have plenty of time to answer any questions that you may have. And so it's my pleasure to go ahead and introduce and turn over the floor to our first presenter, Dr. Chris Marble, Assistant Professor in the Department of Environmental Horticulture, based at the Mid-Florida Research and Education Center in Apopka. Dr. Marble has a research extension split that he utilizes to study and better understand the growth and management of weeds, invasive plant species, and landscapes and ornamental plant production. So with that, floor is yours, Chris.
Thank you, Brian. Thank everyone for having me come speak today. So I'm going to be talking about is creating an integrated research and extension program, and kind of trying to get both of those programs to fit together and feed into each other. So just to give you a brief introduction into me, my appointment. So I'm assistant professor. I joined UF in 2014. I'm at the Mid-Florida Research and Education Center, and my specialty is, like Brian said, weed management and ornamental crops and plants, so in nursery production and in landscapes. And my split is 60 research and 40 extension. And my clientele is really located throughout all of Florida. So the production areas in terms of nursery production, which is where I focus a lot of my efforts, that's really in a few major areas. So all around Orlando, outside of Tampa, and then down, of course, in Homestead. And then there's also quite a bit of production nurseries up in the Panhandle area, up in around Mariana and places like that. So, and then the landscape pest control, really, that's all throughout the state. And then those major markets are just those major urban markets throughout the state: Jacksonville, Tampa, Orlando, Miami, etc. So I'm going to give you an overview today of how I kind of integrated my program and kind of try to give, especially young faculty, kind of at least a template that I used to start my program and then kind of keep it moving forward. And I kind of broke it down into 3 different phases in terms of how I did this. And basically, the first thing I tried to do was assess my clientele needs. So what is the information that they need, both through extension, and then how can my research efforts be geared towards that? So I'm delivering to them the solutions that they need. So the second part will be establishing those research priorities. And then the third part is kind of keeping that whole circle going. So delivering those solutions, and when I'm doing that, trying to seek feedback, so I know how to keep it going and reach my clientele and give them the information that they need. So I'm going to talk about that, and then I'm also going to give some ways that I have tried to use resources efficiently, especially in ornamental plants. Funding can be difficult to find, so it's important to try to, well, when we get funding, to try to use that to meet multiple objectives and have extension outputs and research outputs. And then also I'm going to end with talking about my pitfalls, kind of the mistakes that I make, and then some ways that those could be avoided by some better planning up front. So starting out, so determining where you can help. So the first thing that I did, and I think it's a good idea for a lot of people, would be to kind of conduct a needs assessment. So I did this by meeting with my clientele and with the county agents. So that's one thing that I'm going to stress throughout this presentation is working with those county agents, because they are a tremendous resource and they've helped me tremendously. So when you're meeting with your clientele, developing those relationships and trust, it's got to kind of happen organically, but just investing that time up front, and I think it's going to pay dividends down the road. But then when I met with them and I determined you know, their needs and what kind of information that they need, I tried to figure out where I can use my strengths and my experiences to add value to them. And when I'm adding value to them and I'm able to go out and help them with particular issues, that's another way that you can gain stakeholder trust. So, and one way that I did this was I did a lot of field consultations, also participating in county agent programs. So working with the county agents who give me a lot of opportunities to speak at their programs and meet with their clientele. And then also attending things like EPAP, where you have a lot of county agents together and you can meet with multiple people in one outlet. It's a very efficient way to meet and get to know a lot of those county agents when you're first starting out. And then another thing that I'm going to stress is trying to begin with the end in mind. So trying to figure out how your research and then your subsequent outreach can result in measurable impacts and outcomes, because that's what you really need, is figuring out what you can do that's going to be measurable when you're helping your clients. So ramping up. So first, developing that needs assessment, and then using that needs assessment to kind of set at least some of my research priorities, not all of them, but some of them. So figuring out how my strengths and skill set can benefit your clientele and stakeholders. I think that's a good idea. And then, like I said, again, leaning on those county agents for advice. They're going to be part of your audience and your clientele too. You're developing your program to help them in their duties, but don't just think that they're your audience. They're also an invaluable member of your team and your kind of your outreach team, and collaborate with them and have them on grant proposals and things like that. That's going to be very helpful. And then also getting good survey data. So when you're doing your presentations, your webinars, have polls, things like that. Always work the county agents. A lot of times they'll have polls and things, data that they're collecting from all those programs, but have your own and get the kind of data that you need to help drive those research priorities. And then another thing that I want to point out is when you have that data and you have that survey data from all these programs and stuff, and you know your stakeholder needs, that can be fuel for funding. So you actually have something that you can go to a funding source and say, 72% of my clientele said such and such was a huge need for them. So that's a really good way to get support from funding agencies. And then keeping it moving, so delivering solutions and seeking feedback. So presenting, publishing, posting your research for the stakeholder audience. When you have these research results, it really doesn't do a lot of people any good unless it's published. Of course, that's what we have to do. But presenting it and publishing it in multiple outlets so that you're reaching a lot of audience. Your extension audience isn't going to be spending probably a lot of time reading peer-reviewed research papers. So getting that into a format where it's accessible to them and that they can find that type of information. And then knowing your audience and what they need, how could your research impact their livelihoods and business? So just kind of keeping that in mind and thinking of how they can use whatever that it is that you're working on to improve their livelihood or their business. And then incorporating interactive ways of delivering material to get feedback. So always getting feedback and kind of keeping that loop going. So you figured out what you need, you delivered solutions, but then figuring out what else you can start to work on, and then how you can improve those other research things to just keep that feedback loop going. And then efficiently using resources. So one way that at least I've been able to use resources efficiently, and this came from a lot of advice working with other people, is doing on-farm trials and demonstrations. So it's very common for me to go out to a nursery and I'm trying to help them solve some issue and I don't know the answer. And so I say, I don't know, let's find out. Let's conduct a small trial or demonstration and we'll see what works. And you can set these up to where they're repeated and replicable and randomized so that you can get actual research data from those. And when you're doing these on-farm trials, that's also a great way to incorporate field days. Like I said, if you design it properly, you can get peer-reviewed manuscripts out of that. And then you're getting extension impacts all in one. So a lot of times having research at growers' operations, it's also an opportunity to gather subjective producer data that you're not going to get. So you could ask the grower to go out and rate plants or get their input, and that can be very valuable data for peer-reviewed publications also. And then when you're talking about extension impacts and outcomes, at least I've found, and there's data to support this, that growers are often more likely to adopt new practices or new methods of doing things if they're seeing it work on their own farms. So it is a great way to get extension impacts also. And then another way to kind of get extension out of research is all your research papers, you probably have a good EDIS publication or a good EDIS topic somewhere in there that you can pull out and put on EDIS to reach a wider audience. So extension programs, so trying to turn your extension into actual research. So extension programs, when you're doing, whether it's online, in-person, field days, getting IRB approval is a great first step so that you can take some of that data and you can publish it. So another thing to think about as well is if you're developing some new type of program, whether it be online, in person, some sort of hybrid program, a lot of people could probably benefit from that as far as how you organize that and develop that. So you could think about doing something like an extension methodology paper. Also, when you have that IRB approval, you can have those extension outcome type papers where you have the survey data And then, of course, with these extension programs, you're going to have those outputs and impacts too that can be reportable, and it's measurable things that you can include and show your impact that you're having. So this is just an example of how one project where we were able to really stretch those dollars, I guess. But we figured out that a lot of clients needed non-chemical methods of weed control. And so myself and then Sean Steed, who's an extension agent, we were able to get a on-farm research on-farm grant, basically. That was about $15,000, so not a whole lot of money, but we did that for a mulch evaluation. So deliverables for that, easy to do a field day, had people come out, see it, talked about the results. He also was able to do a video with that, and then Edith's publication on just basically non-chemical weed control methods. And then also when I'm doing these grants and things like that, and when you're working on grants and you're trying to gather up the information in that previous the previous work that's been done, you might have enough to do a review paper. And then also you could do a paper, possibly add maybe another small project onto the side so that you have enough data to have a good peer review paper. And then those, again, with those extension outputs and impacts, you could probably gather enough data with that, and then maybe some follow-up data to see the growers' perceptions, and then how these different programs and these different methods affect their practices. So avoiding my pitfall. So of course, like a lot of young faculty members, I've made plenty of mistakes. But one of the big things that I did when I first started was trying to do too many projects and just kind of doing those adequately instead of focusing on those more important, kind of more impactful projects and doing those exceptionally, or so basically stretching myself too thin. So that's just going to kind of be a decision that you'll have to make. But I would say doing some things very well is probably a lot better than doing many things just, okay. And then also considering your overall time commitment. Florida is a long state, like I like to say, and I have over here on the right-hand side of the screen, you want to avoid this where you have 5 different on-farm trials stretching from Bristol, Florida down to Homestead. So enterprise points are nice, but they're not that nice. So you want to be mindful of your time. And then possibly say very complicated or expensive experiments for controlled conditions, so not on farm. One of the problems I've had, when you're going and traveling, schedules change and things like that. And if you have a very complicated research project on farm, you might not capture all that data that you really intended to. And then another thing that I think is very important is don't just focus on more extension activities or projects to get small amounts of data or feedback from those, but focus on getting more or better data so that you can get more impacts, more outcomes. You're gathering more data per program so that you can really focus on those programs and make those exceptional. And that's all that I have. I know we'll have some time for some questions and answers at the end. That's my contact information. And again, I'm happy to help anybody that I can or answer any questions. And thank you again.
Thank you very much, Dr. Marble. I think the advice you gave was phenomenal and very helpful to people that are trying to really marry their extension and research programs. Thank you for sharing that. We'll go ahead and move on to our second presenter. Dr. Grady Roberts, professor in the Department of Agricultural Education and Communication. Dr. Roberts has a research teaching split that he utilizes to focus on global education, experiential learning, and teaching and learning in university settings. So with that, I will go ahead and turn over the floor to Dr. Roberts.
Sure. Good to be with you guys today. Chris, if you could quit sharing your screen. I've not prepared any presentation this morning. I wanted to keep this more conversational. And so I've really separated my points I wanted to make this morning into 2 things. One is a little bit about me and how I got to be where I'm at. And then the second is some advice that I've picked up over the years related to how to integrate your programs. So I'm a professor of agricultural education, and I hold a 50% teaching and 50% research appointment. I was promoted to professor in 2013. I began my career at Texas A&M as an assistant professor, and then came to UF in 2008 as an associate professor. Since reaching the rank of professor, I have also served on the IFAS promotion and tenure committee, and that's an opportunity I think that some of you that are full professors might want to consider if that comes across, because it was a pretty insightful, eye-opening experience. Also, since making the rank of professor, I've had the opportunity to review dozens of packets from colleagues across the country that are going up for promotion. My research focuses on identifying effective practices for delivering educational programs, and my teaching program focuses on basically teaching people how to teach. I mentioned earlier that I'm 50% teaching and 50% research, and so I don't have a formal extension appointment, but the extension types of activities that I do focus on working primarily with CAL's teaching faculty and IFAS extension faculty to improve their teaching approaches. So some advice I've picked up over the years, and I hope that this will generate some good questions, and we can circle back around at the end of our time together to kind of tease some of these out. Number one is look for the natural connections between your teaching, research, and or extension programs. And if you don't see any natural connections, then that's probably a signal that something's out of alignment, because there should be some natural connections. Also related to that, as state faculty, most all of us have two-way splits, and some of us have three-way splits. You've got to look for ways to synthesize across those, because if we devoted 100% effort to each of those, before long we're giving 200% effort or 300% effort, and that is not sustainable at all. So look for the natural connections. The second point I want to make, and Chris actually brought this up in his presentation, is that scholarship can be generated from your teaching program, from your research program, and from your extension program. And so we often think about generating scholarship from our research program, but we may not think about our teaching or extension programs. And so if we're going to implement teaching programs and implement extension programs, collect some data to validate and figure out what the outcomes of those programs are. And then that data can then be used to circle back around and make informed decisions about modifying your extension and teaching programs. And there are lots of different kinds of journals where that kind of work is publishable. And so particularly if you've got larger teaching and extension programs, think about how you might integrate that into what you do. The third point, and probably the point that has been most beneficial to me in my career, is building teams across your teaching program, your research program, and your extension program. And building good teams is going to allow you to be much more impactful in your work and generate much more scholarship than you would ever be able to do on your own. And so these teams could include, students, both undergraduate and graduate students in your teaching program. It could include stakeholders in your extension program. And I like the model that Chris shared with that feedback loop, because that's exactly how all that could work. You should also think about integrating your grad students and postdocs across particularly your teaching and extension programs. I suspect that most of you guys have your grad students fully integrated in your research program, same as your postdocs, but you may not have thought about how to also integrate those folks into your teaching and extension programs. And then finally, build faculty networks of people that are working on similar things as you, both within UF and outside of UF. And those faculty teams can also be very helpful in helping you keep things going. My next point is, at the end of all this, we have to submit a tenure and promotion dossier, both for promotion and tenure to associate, and then promotion to full professor. And so within your dossier, you've got to be able to tell your story, and tell your story as a faculty member and what your expertise is. And notice that I said story as in singular. If someone's going to read your packet and they're going to, if you teased out your extension program, your research program, and your teaching program, took your name off, and if somebody couldn't read those and think that it's the same person, then you've got a disconnect in what you're doing. And that's a signal that you probably need to think about how can you better integrate some of that together. And then the final piece of advice I have is, particularly as young faculty, sometimes we get really caught up in chasing that really prestigious grant and getting that publication in the journal with a really high impact factor. And those are important, but if we devote too much effort on those sorts of initiatives, sometimes we forget to pick the low-hanging fruit, so to speak. And so look for the pieces of research related to teaching extension and your regular research program that are easily adapted and easily conducted in a timely manner, and get those knocked out of the way. And then once you've done those will often lead to some of those bigger, more impactful pieces. So I will stop there, and we can listen to our final panelists and then bring this back around at the end for lots of great questions. Thank you, guys.
Thank you so much, Grady and Chris. Our third speaker today is Dr. Jose Dubow. Jose is an associate professor in the Department of Agronomy and has a research and extension split. He utilizes this focus on nutrient cycling and forage production systems with an emphasis on carbon and nitrogen cycles. So I'll turn it back over to you, Jose.
Thank you very much for the introduction. And again, it's a pleasure to be here today. So yes, I'm here at North Florida Research and Education Center. I'm associate professor, and my split is 70% research and 30% extension. I also do not have a PowerPoint presentation, but I did take note of some important points that I would like to highlight today with everyone. And one of the things is that you need to be strategic when designing your programs from day one. And what I mean is that, for example, in my case, I have a research and extension appointments. So it is important that one program complements the other. Like, the previous panelists already mentioned that, because they provide feedback, and supply information from one the other. For example, my research program supplies information for my extension programs, and the contacts that I have with the stakeholders, they also give me insights for my research. So this feedback loop is really important. It's a win-win scenario. And like Dr. Roberts were telling, you need to know the rule of the game, for your professional development, for your training and promotion, and play by the book, and at the end, in most of the cases, this process is very fair, and they recognize the successful professionals that you have here. So make sure you check the boxes. For that, I mean, it's important in your strategic view to have long-term goals. And I like to think in using a system approach. In my case, I work with livestock systems, and forage is a component of that. So I'm looking to develop more sustainable livestock systems. And for that, that's a big goal, which, I'll probably get to that point, after a few years of programs. But then make sure you also have short-term goals. And it's important to have those short-term goals, and that would feed, your long-term goals. And at the end, you also, get publications and EDs and extension products and things along the way. And those things will help you to achieve your long-term goal. So in my case, I work on forages, and it's kind of tricky to have, find funds to do applied science on forages. So you need to come up with some ideas in order to build a program that is locally relevant, because that's very important for your stakeholders, but also globally relevant, so that you're generating some science that is important for a broader audience, and that will also help to leverage support from different groups. For example, if you're developing system for the state of Florida, which is our first goal, I think, so that would be important to leverage support from local stakeholders and benefit the state of Florida. And that also will help to facilitate getting funds from commodity groups and from the Florida state agencies and things like that to support your program. At the same time, the global relevance, if you're able to make that connection, the global relevance will enhance the opportunity to leverage funds from national an international agency, which is very important, not only because of the funding, but for your professional recognition. So I'll give you the example of my program where we have a goal to integrate forage legumes into livestock systems. So this is important for producers in the state of Florida because you reduce the fertilizer costs and increase livestock performance and things like that. But it's also globally relevant because you reduce nitrate leaching, reduce greenhouse gas emissions, perhaps increase carbon sequestration. So all of those things, they have global relevance. So if you're able to make those connections link, those two things, so you're able to make your program more relevant for different audiences. The international component is really important as well for your professional development. And at some point in your career at UF, they're also going to ask you for that. So that's very important. It is also important to do the needs assessment, like Chris mentioned. So you need to meet your clientele, understand their problems, and make sure that it's a bottom-up approach, right? So and also understand that I know at the end we're producers are our end goal, but the extension factory, I think that you have located across the state in the extension office, they are our prime clientele, in my opinion, because they help us to reach the producers. For example, Florida has 15,000 beef cattle producers. I will not be able to reach them all. So we need to make sure that we understand the extension system and make sure we communicate well with our extension faculty. In terms of engagement of stakeholders, besides the traditional extension events and extension publication and so on, I found that social media is also important. It's a great way to reach out more people in a timely manner. Sometimes I post like pictures of a new forage variety or new management practice or something like that, and I have producers and extension agents calling me the next day asking more information about those things, and it also increases the outreach a lot and faster, which is important, and also helps to get your program recognized not only in the state of Florida but also nationally and globally. A lot of people from different countries, they also are looking into that. So, but you need to be strategic on that. You need to engage with the right groups and and try to. And for me, I consider myself almost like an old timer. So you kind of need to reinvent yourself and get into that new world and be strategic about that. So I think UF in general is doing really well on that side, and I see a lot of programs doing a lot of things in that area. And this year is a good example for that, where because of the pandemic, everybody had to kind of reinvent themselves and learn better how to use Zoom. I use Zoom for a long time because I'm here in Mariana, and to communicate well with everybody, I need to use Zoom. But it looks like today everybody have learned really well how to use Zoom, and that's important to connect. And sometimes we have, lives and things that are going on this year that we have even more people than if we had, the physical presence events. So make sure that you try to publish your extension materials in outlets that are important for your clientele as well. And that includes, besides your, scientific peer review publication in your, top journals of your area, you also need to reach out to the industry magazines. For example, we have here the Florida Cattlemen Association, that they have a magazine or others, and that's very important, to reach out to clientele. In terms of practical tips, always engage with extension faculty in a very collaborative manner. That's very important. Sometimes if you don't know a particular answer, that's okay. Just tell, well, I'm taking a look and I'll talk to other colleagues and get back to you. But make sure that you follow up on those things, not only extension faculty, but also with producers. This is very important to follow up on things and not leave it behind. And try to measure impact since the beginning. And for that, that's important to have SMART goals. So it is very important to have measurable results. And like I said, since the beginning, you need to organize your programs, research and extension, put objectives, but make sure that you put realistic objectives and that you can really measure them along the way. Because again, you're going to be asking for that when you're going for promotion and tenure. Teamwork, collaboration, it's key both for research and extension. You know, without collaboration, it's not going to, I'm just being very bold on that. But make sure that you pick good collaborators. Not everybody is a good collaborator. Make sure you pick people that you help your program. You're also helping that person, but you know it needs to be good for both sides. And something important for me as well, specifically my program, is to have a good recruitment pipeline. So I for my graduate students, I like to bring first them as interns, and then they stay here for 6 months, and then some of them we retain as graduate students, some others we don't. And that's important so that you recognize ahead of time, the potential good students to work on your program. Well, I think that's what I have for now, Brian, and I'm sure you have questions towards the end, and I'll be around here if somebody has any questions.
Great, thank you so much, Jose. Appreciate that. And this, I guess, allows us to transition into questions that you folks have for our panelists here today. Anne, do you want to go ahead and pick your favorite so far?
Absolutely. Thank you, Brian. We have a couple really broad questions, so why don't we start there? The first one is a question asking, could you give a few examples of how I can integrate students into my teaching program?
I'll take a first stab at that, and then anyone else can jump in that has anything to add. And so I could see that happening in a couple of ways. First off, if you have graduate students and postdocs, then they could be brought into your classes as either teaching assistants or deliver guest lectures, things along those lines. You could also involve your graduate students and postdocs if you were doing any educational related research embedded into your courses. And that might be an opportunity for them to get experience with a different kind of research than they might do in your lab. In terms of undergraduate students, you could bring undergraduate students into the courses also as teaching assistants, different roles as in graduate students, and they can either do that. Most departments have like an internship credit or a teaching assistant credit, and so it could either be done formally through that process, or it could be done informally in terms of however you wanted to integrate that. But I would also flip that now backwards, though. How can you get your students, other than your graduate students, but the students out of the courses you teach, involved in your research and extension programs? And you might think about building class assignments in that would involve students contributing to data collection, both in terms of advancing their understanding of the concepts in your courses, but also in terms of then actually contributing data that might be relevant to your research program. And I've got an example of a course I'm teaching right now where I'm doing just that. I've got a graduate level course on adult education. And so I have a research project going on right now to try to identify some of the best teaching practices that adult educators use. And so in that adult education class, I have my students as a class activity, each going out to observe an adult educator, interview that person, take some notes about what they see when that person teaches adults. And now the people they select to participate for their class assignment are not obligated to participate in my research, but my students now have the ability to invite the person they observe to participate in my research, which would then expand data that I would get out of that process. So that's how it's worked for me. I'd be interested if anybody has any other ideas.
Dr. Marable, Dr. Dubow, anything you'd like to add?
Well, I do not have a formal teaching program, but I see different ways that it could potentially integrate students in the teaching program besides the graduate assistantships and teaching assistantships that they could help in the program. They could organize, different activities with your programs. In my particular way, I use my students in the extension program, which is a kind of teaching for the, for another clientele. But we've developed, different content materials, and they help me in the field days and extension talks and different activities. So that there are multiple ways to integrate them, I think.
Okay, and I can see in the chat that Dr. Brun is also going to address this in his session later today. Another question back to Dr. Marable, our first session, Chris, when you were talking about how to prioritize and where to focus. What advice do you have for faculty on methods to prioritize your projects so that you can achieve the best results?
As far as research projects, I guess, or extension?
I think research primarily.
Yeah, so maybe it just goes back to that that needs assessment type thing. So people probably are coming in already having an idea of the type of research that they're going to do, or just their knowledge of the field and some knowledge gaps that they can probably start to address. But then when you start to get more specific and you develop those relationships with the stakeholders, and you're talking with agents throughout Florida and then really throughout the region and the country, you start to realize those needs, those questions that people have, and those things that the industry, whatever industry someone is struggling with, that they could potentially address. And so a lot of times with a lot of things, at least in my area, there's a central theme that is coming through, like for weed management. The big thing over the past, I don't know, 5 or 10 years in nursery production specifically is labor costs and labor availability. So there's a lot of different things that could be, geared towards that. But then I'm trying to focus a lot of my research efforts on that in terms of ways that growers can reduce their labor costs and get more done with less labor. And then that's also going to be a good source for funding, because those funding agencies are probably familiar with those priorities as well.
Thank you. We have another broad question. How valuable to tenure and promotion, this is an opinion question, are the internal funding opportunities?
I can start us.
Yeah, go ahead, Grady.
I can start us out, and the others can chime in. And so, we need funding to be able to do the types of programs that we do. We had a past IFAS research dean that used to say frequently that, but funding of any kind is on the input side of the equation. What's most important is the output side of the equation. And so if you look at someone's body of work, if they are adequately funding grad students, if they're adequately generating research publications, if they're adequately having results that are deliverable to stakeholders, that's an indication that they have adequate resources for their program as designed. If the person is having some deficiencies in those areas, then perhaps the reason is lack of funding. Now, specifically about internal funding, oftentimes, particularly some of the bigger funding agencies, they're going to want kind of a proof of concept, or at least some initial results to show that what you're proposing has potential. And so oftentimes those internal funding pieces can then allow you to collect some initial data that would then allow you then to extrapolate that and go after additional funds from the outside.
Yes, and that's very important. And I would say take advantage of what you have here at UF in IFAS, because there are amazing opportunities for early career grants, the equipment grant, and I can tell those things help me a lot, to build my program. And like Reddy said, they will help you to generate, preliminary information, and that to be important, to seek bigger grants later. The equipment grant, allows you to, build your equipment infrastructure and help as well, to move your program further. So I think those are the main things, they help to establish your program, and and there are great opportunities, of funding here. So take advantage. Not all universities have all these opportunities that you have here, and make sure that you take advantage of them.
Yeah, and I would just say, I would echo exactly what Grady and Jose said. That it's a great way to start your program, and then use that as use that to develop your baseline data that you need to go for those larger external sources of funding. So, but yeah, the outputs are going to be the most important thing, I think. So.
This is kind of a follow-up question that Dr. Maribel addressed a little bit, but could the three panelists expand upon how you can utilize stakeholders, essentially, their voices to fuel funding and the need for funding?
Basically, I mean, like the way that I would try to I did that was like I was talking about, with gathering those that survey data and things like that. And even if it's not something that I'm going to design the the survey to publish it or anything, at least I have something when I'm writing a grant proposal that I can say I have data showing, from 100 and something nursery growers in Florida, and 85% of them are saying that labor costs and labor availability is their number one concern, and weed management is taking up, such and such amount of their labor. And so at least I have something that I can go to those funding agencies and say, I know that this is a need because I have growers that are saying that this is a need, and I have data to show that. So that's one way that I did it.
Yeah, I would echo what Chris said. But if you're going to collect that data, my advice is, though, make sure that you collect it in a way that it is publishable. So then you actually generate some scholarship out of that piece as well. And so then you're getting scholarly outputs all along the way. And the funding agencies, particularly the federal funding agencies, I mean, they're designed to address the needs of the people, right? And as state employees, we're supposed to be addressing the needs of the people. So if we can go beyond just our own opinions and show documented things for stakeholders, and then depending on the type of research you're doing, you might very well need stakeholders at some point in the process, either for field trials or for implementation, or at the end to evaluate the efficacy of what you've proposed. And so having those linkages all along and showing that it's not just this disconnected group of stakeholders, that you've actually involved the stakeholders in kind of conceptualizing the research project, and have partners in there at some level that show how you're linking with stakeholders, I think is important.
Yeah, in my case, I interacted a lot with the Florida Cattlemen Association. They're a very well-organized group, and they do have a research committee, and they, created, based on the group, their needs. They have a list of the top 10 priorities of, research priorities for that particular group, and they're broad enough that you can put your research in some of those things, and that's very helpful, actually, to justify some of the research that we do.
I think one other comment about stakeholders too is that, as you put together your proposals, you have the option of defining the stakeholders and the beneficiaries in the way that you see fit. And if, depending on the nature of what it is you're trying to do, if you define it as a very small group of stakeholders, then the potential impacts of your work are much more isolated and may not be as impactful. Whereas if you could step that back just half a step and define it as a larger group of stakeholders, then the funding agency might say, wow, this has even bigger potential for impacts than just this small group.
It's excellent input, thank you. Another question building upon that, in particular for faculty that are located in Gainesville, kind of best practices for establishing relationships with stakeholders in various communities?
I guess I'll start us out on that, since of the three of us, I'm the one based in Gainesville. So yeah, our beautiful campus in Gainesville can be an island if we're not careful, and we can get stuck on that island and never go out. But we've got a tremendous asset in IFAS, and that IFAS has a presence in all 67 counties of this state, in terms of the extension offices, and then scattered throughout the state in terms of the research and education centers And then most of your departments have faculty on many of those research and education centers. And going back to the point I made earlier about building those partnerships, if you're out in the state and if you build those partnerships, then that avenue can go back and forth, because there's some ways that we have advantages on the main campus in terms of resources and inputs, but then there's also some advantages that faculty at the RECs have and in the extension offices have too, related to their being located in a setting that might be very useful for our research, extension, and education programs.
A very good point. And we do have 12 research and education centers across the state, plus the 67 in our extension office. So there are numerous opportunities to interact with those folks. And that's true also for the REC faculty, to interact with the gains with faculty and vice versa. And we've done that, we don't have any problems in the issue. A lot of, people from gains who come and work together with us and
Thank you. We are nearing our end for this session. Are there any additional questions?
I'll throw one out there. Jose talked about a balance of short-term and long-term questions, and I only have a few minutes left, but I'd be interested to hear the opinion of our panelists regarding how they've gone about their methods for balancing that short-term and long-term goals with some specific examples.
I can start that if you like I said, I like to think as a system approach. In my case, livestock system, sustainable livestock system, which has a long term, to develop those things. But then to fit those things, I have something in mind. Okay, I need some legume varieties. I need some particular practice of, some type of new fertilizer or whatever. So I do several different trials, and they kind of come up together by whenever, developing that bigger sustainable system. But I need those multiple pieces that I'm generating in the short term. But in the long term, I'll put them together to get where I need to get. So and that also helps you, along the way, because you cannot wait like 7-8 years to have a product. You need to have multiple products along the way. So that's my perspective on that.
Yeah, I would just echo those thoughts as well. When you're designing your research priorities, it should be especially when you're starting out like me, assistant professor, you've got to have those publications, and so you can't wait 4 or 5 years for a study to complete. So you have those longer term goals and those longer term research experiments and those objectives. But then along the way, you have these multiple research projects and experiments and things like that, and then together those kind of those short-term goals will lead towards that longer term, the big picture type goal. So depending upon whatever field you're working in, that's going to look different, but that's just kind of how you would structure that, I guess.
Yeah, and I would echo that as well. Think about the bigger system and how the little pieces that you are working on impact the bigger system. And then something I recognized early in my career that I was not doing a good enough job on was helping my graduate students see the bigger picture as well, right? Because they're only with me for a short period of time and tackle one little piece, but they don't see the bigger impacts. And so that goes into, like I said earlier, about telling your story in your promotion and tenure dossier, where how does your little piece fit in the bigger research question?
Excellent. Well, I'd like to, that concludes our session here today. I'd like to thank our panelists for being here and sharing their knowledge and experience with us. I'd like to thank my co-moderator, Anne, as well as Dr. Adams, Dr. Gilbert, and Cassie for making this possible. Our next session begins in about 10 minutes at 10 o'clock. So thank you again for everyone's participation.
Hey, good morning, everybody. Thank you for joining us for this next session. Hopefully everybody can hear me. I'm going to be leading this next session, which is managing large and complex research projects. And so the goal of this session is to discuss how to manage college, interdisciplinary, and multi-institutional projects by some of our faculty experts, and they're going to share some advice on how to manage data, integrate research, and also stay on target to meet project milestones for large projects. And so these talks are going to be a little bit longer than 10 minutes, and they will be followed at the end after all of the speakers have talked by approximately a 10-minute question and answer period, much like the sessions prior to this. So please, if you have any questions, add those to the chat and we will try to address as many of those as we can. The first speaker today is Dr. Carrie Harmon, and she is an extension specialist in the Department of Plant Pathology. Her research focuses on improving plant disease diagnostics and validation for emerging and high-risk pathogens that are threatening Florida agriculture. So today she's going to talk about supporting multi-state extension support for plant diagnostics. So Carrie, I'm going to turn it over to you.
Unmute, there we go. All right, well, thank you very much for inviting me to talk about this. This is my favorite thing to talk about, is this big project that I've been doing now for 17 years. So I'm going to talk about how it grew, just make this easy, how we got it going, how we've grown it, how we've sustained it, protected it, built it, and then the pieces of what it takes to keep so many people going. This is an extension project, so the rate of pay is not necessarily commensurate with the effort that people put in. And I don't mean personal salary, I mean the amount of effort that people put into this project and the amount of funding provided by the project itself. They aren't equal, so it's a lot of cheerleading. Heavy text, but the main point here is that the National Plant Diagnostic Network is a cooperative agreement. Well, it's actually 6 cooperative agreements, 5 regions, and then a data center. It was established in 2002 as a response to biosecurity concerns post 9/11. And at the time, the USDA CS-REIS was our agency. Now it's USDA NIFA, the National Institutes for Food and Agriculture. Most of us in the extension side of things and in IFAS are very familiar with USDA NIFA. I want to stress that it has always been a cooperative agreement between NIFA, the federal agency, and the home institutions. And that it's not just a grant, it is a cooperative agreement, meaning the government provides guidance and support, and the institution also provides support and activity. Each regional center, and I'll show you sort of a picture of what our project looks like nationally, each regional center manages 9 to 13 subcontracts. That's 9 to 13 states or territories. There is 1 per state or territory. But it's built on the existing land grant extension system. So there's a diagnostic lab in every state and territory, nearly so. And most of these are plant disease labs, but we also have several other labs. So Billy Crow here at UF, his nematode assay lab is one of those. And then we have 5 pathology labs across the state. And the insect ID lab is one of our labs. So within Florida, we've got a FPDN, a Florida Plant Diagnostic Network. Then there's a regional Southern Plant Diagnostic Network, which is actually the project that I am the PI of. And then there's the national project, which at the moment I'm the executive director of. So the project itself was initiated with an emergency funded 2-year seed project. And it was basically, here's some money, get the best minds together and build this thing. And that's when I was brought in, and I was funded completely on this project to help get it going. So at the time, those projects all went to department chairs because those are the people that had power to allocate personnel time, funding, that cooperative part of the project. It is no longer only department chairs that can hold the regional projects. I'm an example of that. I'm not a department chair, but I am the PI of the project. But at the beginning, we really needed that piece. So the take home out of getting this huge project going is that you've got to find people who are willing to put in the time, because the amount of money isn't always what makes the difference. really it's the amount of energy people had to contribute. So this is what the project looks like graphically. It's a hub and spoke system. So every region, the orange is the hub. That's my lab, my program here at the University of Florida. UF is the hub. The IFAS extension system is the hub for the whole southern region. That includes Puerto Rico and the US Virgin Islands. Then there are 4 other regions and our data center at Purdue. So growing, when you're trying to grow a project, when you're trying to say you've got a multi-year cooperative agreement, so you've got some time to get going. This isn't like a 1-year quick research project. Remember, this is an extension project. We're growing connections. We're growing communication between people. So right now we run somewhere between 3 and 4 million a year. There have been times when we've gotten more, times when we've gotten less. The amount for my project over the last couple of cooperative agreements has been around $560,000 to $580,000 a year for the whole southern region, with 13 subcontracts to fund. In 2012, thanks to IFAS, $4 million worth, my program got a new building, the Plant Diagnostic Center, which is the kind of proof that we're looking for at the centers to show the cooperative nature of these projects. So UF really IFAS put up real support, substantial support, not just in allocating personnel time to it and supporting the grants and the stuff that our IDCs, our indirect cost fund, but really said, you know what, the little tiny building with the ceiling falling in, that's not going to be good enough to house the regional hub of this huge project that's got a lot of recognition. We're going to do something bigger. And we got a new building, which is the only time I imagine this will happen in my life, certainly in my career, and it was astounding. We are still appreciative of the program's housing in the little brick building behind Fifield Hall. The other thing that IFAS did was take on my salary. Up until then, my salary was, I brought in all my own money on all my research and extension projects. The grants that I did guided what I needed to do for my daily job. And IFAS decided that having my position to direct the diagnostic lab, which is the hub lab, to direct projects like this and to leverage funding, I had a proven track record of success, and I was probably still a good investment. I hope I'm not putting words in anybody's mouth, but I think that was sort of the idea behind putting me on state dollars instead of full grant funding all the time. My grant dollars have gone instead to hiring more people to do this project. So it's not that I've stopped pulling in salary dollars, it's just that money is now used for other people. We're in the final year of our 5th multi-year cooperative agreement for this project. I've been the PI for the last 3 cooperative agreements. We're applying for the next one, spring 2021. It will be competitive, so technically the hub could go to any other institution in the region. The chances are not good that it'll leave here because of the amount of support that IFAS has shown to this project. There are probably no other institutions that are able to say they've done what we've done. And by that, I mean we, as in the administration of IFAS, supporting this project. Across the whole network, though, nationally, there are 72 active labs, 5 regions, and 1 data center. The program members are diagnostic professionals. So these are extension people through and through, most of whom do not only the diagnostic work, or that's our extension service, but they also participate in teaching. They help to do research. These are folks that are completely involved in the 3 legs of the land-grant mission. And program leadership is land-grant university faculty working with our NIDFA national program leader. Stakeholders, because we work through these labs and through the extension system, our stakeholders are the stakeholders of the extension system and our diagnostic labs. And to get all our job done, including the research and leveraging into new projects, we work with IFAS, ARS, our partners in our departments and partners across campus, the IPM folks, the state departments of agriculture, and of course, the industries that we serve. The only way to keep moving this forward is to stay organized. And I'm going to emphasize that if you can't be organized, if you don't have an assistant to help you organize, or you don't have a system in place, managing a large multi-state or multi-cooperative project among collaborators is almost impossible. So how have we survived for 17 years? What's the sustainability of our program? I'm going to talk specifically, we had a budget slash year where we were cut almost in half. It was a year that the economy was not doing great across the country. NIFA didn't have as much money to work with, and we got slashed. And so what we had to do was take a really good look at our whole program. What could we ease back on so that we focus on our core. What is the core of what we do so that we can keep doing that? And these other pieces, we have to find ways to either hold them off for a little while, sort of put them on ice, or leverage them out with another partner that does a similar thing. And we did both of those things. Some projects we just had to stop for a little while. Didn't change our deliverables to NIFA, it just meant we had to condense what we were getting done in a given year. One thing that's been important is impact evaluation. We have to be able to prove, as any extension or research program, we have to prove our worth at the local and national level, because our funding is going to match the effort, hopefully, right? Even though I started this off by saying the funding will never match the energy, we do get funding from the government based on what we can show the public and who is served by the public, our legislators and others. They have to know what we're doing. If they don't know what we're doing, it's hard to justify asking for money. We make sure to invest in the people and the future of the field. So our project is not only sustaining these laboratories in a small way, in a cooperative way, but we're also educating grad students. We've got a training pipeline so that we've got a next generation that's going to be here when the diagnosticians and the faculty who are in these positions now, when they retire, so that we can keep this project moving forward. It continues to be justifiable. And that makes the other piece of that is there's no stagnant. We can't be stagnant. We've got to be making sure we're delivering hands-on training in new organisms, new to me organisms, new technology. And we have to invest in the infrastructure and IT. And IT really is part of the infrastructure. But whether that's in our home institution or across the network, it doesn't really matter because we're all drawing on that. So we have moved infrastructure to being not just in the labs, but also what supports everybody at a national level. We had a little evolution and some revolution in our strategic planning. We needed to really refocus. We had to be able to reflect the fact that times are changing, that people's needs are changing, the industry is changing, extension has changed. We have to be changing with it, or we are no longer relevant. And so we went through a 2-year strategic planning process, which was not fun. I'm not going to pretend it was, but what the product is now going to guide how NIFA writes our next RFA, for our next cooperative agreement, and how we move forward with prioritizing projects. So it really is about putting our money where the plan is and implementing on a national level so that we have removed any redundancy between regions and everything goes national. We are emphasizing the diagnostic labs and their network contribution, not just from the labs, but their home institutions. And then we have to turn around and say, well, what is being involved in this project going to do for you? And part of that impact evaluation is being able to demonstrate that participating in this project brings benefits home to the home institution. We have leveraged out leadership of our committees to make sure that non-regional center people are leading the effort so that we make sure we're actually hearing and then implementing what needs to be happening within this project. And we've tried to do our best to make reporting and coordination standardized and as easy as possible for the participants in the project. That means, again, staying organized. So the cajoling, the cheerleading, the convincing, that's my job right now. As executive director, it's a 2-year role. I'm halfway through it. And my job is chief cheerleader for the project, because a lot of what we do is really, it's what everybody wears multiple hats for. It's their extension job, but it's also a lot of volunteer effort. Their time is accounted for, but the energy they put in, you have to make sure it's got a place to go. Make sure that we've got outputs of use to external supporters, our stakeholders, people who use the labs, and make it easy for the co-PIs to participate and report in, and then make it easy for the agency to justify the money for our program. So we've put together, lots of PowerPoints for extension presentations, standardized and shareable protocols for the labs to use, where our job is to save everybody time and spread out the expertise so we don't have to all be experts in everything, because we can't. I know I'm not probably supposed to say that, but we cannot be an expert in everything. We connect the collaborators to each other with national meetings that are focused only on what our field is. We have a diagnostic network meeting every 2 years that is focused just on diagnostics, on connecting our collaborators. And then we've got regional calls and meetings and listservs and ways to make sure that people know what's going on, know how to get involved, and know how they can benefit and participate. And then my job is doing my best to make everybody look good, both at the home institution and then within the network itself. So sustainability means staying relevant. We've got to be able to organize response to needs at the local and the national level. So if there's a major outbreak of a disease or a pest, we have to be able to mobilize and be able to identify that thing if it comes into the lab, to have information to push out to the public. We've got to be able to do our job, whether it's at the local level, something new in Florida, or the national level. We've got to be able to curate our outputs. I don't mean just make them. I mean know what ones need to be made, which ones need to be updated, how we need to advertise them and make them available to people, and when to retire them. So I know we're all used to the EDIS system and the 3-year, 2-year sort of timeline to sunset. We have a similar system, but it doesn't have quite the same timeline to sunset. But we do evaluate everything we've got out there for outputs to make sure that we are meeting needs, not just pushing out stuff. We are looking further outward, not just within the country, but international partnerships, because pests don't respect political boundaries. So something in the Caribbean is eventually going to get to Florida. Something in Arizona might eventually get to Canada. We've got to work together. That again helps us to extend our expertise. We're planning for Generation Next. I sort of stole that from this old house. Both in the human resources and in the technology. You have to make space for new voices and allow transfer of knowledge from those who want to roll off a project gracefully, but without losing that institutional memory. So we don't always have to do it away because it's the way we've always done it, but you also don't want to throw the good stuff out just because somebody retires. And that's only important if you've got a project you want to sustain for many years. Imagine that's lots of projects, but in this case for us, we really want to still be here in 10 years. So this has become a really important focus for us. In the end, I have to always try to say yes. That's the extension thing. Somebody comes to you with a need, you find a way to help them, even if it doesn't actually come from you personally, right? You find who it is that they need, and you be valuable, and you just leverage the snot out of every opportunity because that's how you sustain a program and grow it and expand it. I hope I'm within my time limit. I'm happy to take questions. I'm happy to talk with people long after this is over. I know this is the research forum and my project is extension, but it has leveraged into research and teaching products that have helped both here at the home institution and farther than that. So thanks for listening.
Thank you so much, Carrie, for sharing your experience with that project. That's really helpful. And if anybody has any questions, please do enter them on the Q&A chat, and they will be addressed after our next speaker. So our next speaker is Dr. Fred Gmitter. He is a professor in the Department of Horticulture and is located at the Citrus Research and Education Center in Lake Alfred. And his research program focuses on citrus breeding and genetics for variety improvement, genomics for plant improvement, citrus germplasm characterization, and also applied genetic transformation research, among other things. And so today he's going to talk to us about his experience in managing a large-scale international consortium project. And the title of his talk is Lessons from Global Cat Herding, the International Citrus Genome Consortium. So take it away, Fred.
All right. Well, I'm really pleased to be a participant in the IFAS Research Forum today. When Dr. Peltz-Stalinsky invited me to come to Gainesville in September, I was real happy to have the opportunity to come up and visit with colleagues and spend some time in Gainesville. But unfortunately, here we are in Zoomville, Florida, and we'll just have to put up with that. The title of my talk today is Lessons from Global Cat Herding, the story of the International Citrus Genome Consortium, which is a group of researchers that I've led for several years. I'm usually reluctant to use trite cliches like cat herding, but in the case of this story, cat herding is probably the closest thing to describe what life was like working with a group of international researchers in citrus genome sequencing. For those of you who don't know the phrase herding cats, and I learned from my sons today, they didn't know it. Herding cats can be defined as a futile attempt to control that which is inherently uncontrollable. This slide shows a brief history of genomics, and it goes back all the way to the mid-1800s when Gregor Mendel was playing with peas in his garden and unraveling the secrets of genetic control of traits. What I really like you to focus on is the period of time from 1990 to 2003. In 1990, the Human Genome Project in the US was launched. It took 13 years from the launching of the project to the time when the first human genome sequence was completed. And along the way, there were many steps. In 1997, JGI was established and Genoscope was established in France, 2 national level genome sequencing centers. You'll notice a number of papers published, including papers in Nature and Science. So this is sort of the upper stratosphere of scientific endeavor, at least in terms of where they're published. And what you'll notice, or what I'd like to highlight, is the Human Genome Project, which was initiated in 1990, didn't really have its first 5-year plan established until 1993, 3 years just to make the plan, and another 10 years to actually complete the project and have the first so-called finished version of the human genome sequence. This Human Genome Project, 13 years, cost substantial money, as you can see listed at the bottom. This year, 2003, is very important for us in citrus because as we in the citrus community looked at this wonderful work in human genome sequencing, despite the huge cost, this was something that we wanted to engage in as an international group. Our reason for looking at genomics as the way of the future for citrus is because having good genome sequence information enables us to identify and manipulate genes of interest that control desirable or undesirable traits in citrus to facilitate breeding and genetic improvement. The sequence information also can give us some very specific targeted sequences to expedite the genetic improvement of citrus. We imagined a time where having such information on genome sequence would actually enable growers to have tools to be able to optimize their production practices by looking at how different practices might influence gene expression and understanding what those genes might be. And of course, in the end, we saw huge potential benefit to consumers by work we could do with this new understanding to improve citrus fruit quality, both the flavor and the health-related benefits of citrus fruit. The requirements for whole genome sequencing partnership are more or less listed here. We had a target of covering the genome 8 to 10 times over with sequence, which was a typical level of coverage, using Sanger sequencing technology, which at the time was the gold standard that was used for producing genome sequences. It was a very slow and very expensive process. And at the time that we began our consortium, we estimated we needed somewhere between 3.2 and 4 million US dollars to be able to accomplish this. Being citrus researchers and ambitious, we wanted to begin this as soon as possible. We had plans in place for the rapid deposition of data that would be allowed to be freely accessible to all partners in the consortium, and eventually to be made public to all the citrus research community. We used common libraries, plans in place to share and distribute resources, and so on. Our rationale for an IGC, the most important one is listed there at the top. The magnitude of the tasks at hand to do this exceeded any one group's capabilities and indeed any one group's budget. Remember, we were talking about 3 to 4 million US dollars to do this. And at the time, the Human Genome Project was accomplished by some of these major national level, international level sequencing centers that focused on cranking out sequence. a greater part of our rationale as well was to develop genomic tools that could be used freely available to all citrus researchers worldwide. So a huge contribution to the overall scientific research programs in citrus. We knew that by working together as a consortium, we would be able to foster greater research cooperation and collaboration and encourage sharing of resources, information, and ideas. We selected a kind of sweet orange called the Ridge Pineapple Sweet Orange. And this is the first example of the role of politics. I'll discuss that in just a moment. So in 2003, a group in Spain managed to cobble together some money to invite global participants to Valencia, Spain, to discuss the possibilities of putting together this consortium. At that meeting, we spent several days together, and at the end of the meeting, we issued what was called the Valencia Declaration, a very European type approach to things. We have a declaration to make, but it stated quite clearly what it was we hoped to do and why we wanted to do it. And this was followed up shortly thereafter by a white paper that was written collectively by the group. The purpose of the white paper was for all of us to have a document in hand to go back to our respective industries, our granting agencies, our institutions, and point out that we were a small part of a larger international effort in hopes that we could pull together funding from various sources to meet our needs to actually pull this ambitious plan off. The initial partner nations are listed here. At the meeting, there were representatives from Brazil, from China, France, Italy, Japan, Spain, USA, including myself and others from the University of Florida. There were several other nations who participated in the meeting, including South Korea, Israel, and South Africa. There are some countries highlighted with asterisks here. They were the group that finally stepped forward and actually became involved with the sequencing project itself. So we had beautiful plans. We had a number of cats all together, but where, oh where did the cats go? They went hither, thither, and yon. This lists the countries and the scientists who were the lead people in these countries who actually remained within the consortium. Very early on, our friends from Japan and from China informed us that, in fact, they weren't able to participate. They didn't see a way that they could get financial resources. And at that point in time, essentially, they left the consortium. We also partnered with a number of these large national level genome sequencing facilities. Genoscope in Paris, France, IGA located in Udine, Italy, the US Department of Energy Joint Genome Institute, particularly Dan Roxar, Simon Prochnik, and Albert Wu, underlined because he's been a key player, a key cat in all that we've done over these years. Hudson Alpha, which is a satellite of JGI located in Alabama, was involved. And a private company, Life Sequencing in Spain, likewise was involved. So this was the partnership of citrus researchers and genome sequencing facilities. To come back to Ridge Pineapple Sweet Orange and our first political challenge. When we had the meeting in Valencia, we wanted to select what would be the citrus plant we would sequence. Our friends in Spain wanted to sequence Clementine mandarin, it being the most widely grown and most commercially important variety in Spain. Our friends from Japan wanted to sequence the Satsuma mandarin, Satsumas being the premier culturally relevant, culturally important citrus fruit in all of Japan. The Chinese wanted to sequence Pongkan mandarin, which is the major mandarin grown in China. Those of us in the US and Brazil thought, well, we should be doing sweet orange because sweet orange is the most important. So we had our first big political discussion. And in the end, we worked through it all and we concluded that we would sequence a sweet orange because even though there were local political reasons in other countries to sequence other things, everywhere in the world that citrus is grown, sweet orange is also produced. So it was the ubiquitous citrus for us to work on. Right around this time, JGI had published the poplar tree genome, and they encountered substantial difficulties in putting together a quality genome assembly. Poplar, just like citrus, is a plant that's highly heterozygous. It has usually 2 different copies of a gene at the same location throughout the genome. And so the assembly of the genome becomes incredibly complicated and prone to error. And they asked if we had any haploid citrus plants, haploids having just 1 copy of each gene region. And in fact, we did. We had several. And together, the group evaluated these 3 on a number of different criteria. And in the end, even though originally we selected Ridge Pineapple Sweet Orange, we settled on a haploid of Clementine Mandarin from Spain because that was the best material for us to begin our exercise. However, Ridge Pineapple did remain in the story. But the question came, who has the money? We met it first in 2003, and already we were in 2008, and no one had put any money on the table. Our grand plans were becoming just a grand plan that never came to fruition. Time quickly passed. Unfortunately, in Florida, we were faced with citrus greening disease, a disaster to our Florida citrus industry. And many of us recognized that if we had a genome sequence available, that we could actually, in fact, use this for all sorts of research that might help to address the problem of citrus greening. So I asked the Florida Citrus Production Research Advisory Council for $500,000 as a catalyst to put some money on the table to see if we couldn't stimulate some of the other countries to step forward. I can still remember the day that they funded me, and they gave me $250,000, not 500. And I can recall a citrus grower coming up to me after the meeting and saying, "See, Fred, we only liked you half as much as you thought we did." Nonetheless, we had $250,000 on the table. I immediately contacted my colleagues around the world, and soon everyone stepped forward. The French had money. The Italians said they would find a way to do it. The Brazilians got some money. The Spanish got money, and soon we were on our way. It took 8 to 10 months to accomplish this from the time I got the $250,000. And being a University of Florida employee on a 1-year grant, I was about 6 weeks or less from the end of the grant period with $250,000 in my pocket. And I wanted to make a down payment at JGI, just telling them we'd get more money, but JGI could not accept a down payment. So I was faced with the dilemma of how to wisely spend $250,000 in 6 weeks or less without violating any of the rules and regulations of the university and the grants office. So I contacted my friends at JGI. They told me about a new startup company that was called 454 that had new sequencing technology. And they were looking for poster children to demonstrate the value of their much less expensive technology. So I sent an email to the vice president of the company, and in 10 minutes, I had a phone call back asking me what was the situation. And I told them about sweet orange and HLB, or citrus greening. And before very long, they had agreed graciously to accept my $250,000. They sold us $250,000 of sequencing kits, but they also were huge contributors on their own. They sent a sequencer, a 454 sequencer loaner to UF's ICBR group in sequencing that was dedicated solely to this project. And in fact, they were involved with a lot of the bioinformatics and the assembly and the analysis. However, this presented some problems when I went to meet with my colleagues to tell them that not only were we as an international group sequencing the Clementine genome, but here in the US, in Florida, we were also sequencing the sweet orange that we started out to. And there was a lot of political pushback. I heard stories about typical Americans always running on their own, doing things on their own, never learning how to work as a community, et cetera, et cetera, et cetera. So there was a challenge. Things proceeded, and Florida finally agreed, the industry, to support 2X coverage, and all the other things that we had in line were ready. We sent DNA to Genoscope, to IGA, and to JGI. And what to do with that transatlantic rift? It took a lot of diplomacy to convince my colleagues that, in fact, We weren't trying to run ahead of the consortium. In fact, what we were doing would be a lower quality genome sequence, but it would be a part of what we were all doing together. We would include it all as one. And in fact, in the end, we were successful. We produced 2 citrus genomes, and they were made available simultaneously, globally, in January of 2011. It was the first citrus genome sequences available in the world. Being researchers who are not only excited about having a resource for others, but also wanting to put a manuscript together and getting some academic recognition for what we had done, we had a manuscript in process. It included both genomes, and everyone who was involved with the entire project was included as a co-author. So we were working along trying to get our paper organized. In the meantime, I visited an old friend of mine from Huazhong Agricultural University in China, who had pulled out of the project initially, and he informed me, in fact, that their group was doing their own genome sequence. We had a very frank and open discussion. He told me what he was doing. I told him what we had been doing, and as old friends, we agreed that we would publish simultaneously. And as a good faith step, I convinced the international consortium that we should share our manuscript with our Chinese colleagues, and they would share theirs with ours so we could see what each other had and we would publish simultaneously. Unfortunately, that did not come to pass. There were some political forces, I think we might say, that led our Chinese group to continue to proceed along their own way. It was a huge disappointment. And I took some heat from all of my ICGC colleagues for being a trustworthy leader and essentially not getting what we wanted. So what happened? Well, we were scooped. There was no question about it. In November of 2012, our friends in China published a paper in Nature Genetics, a very prestigious journal, titled The Draft Genome of Sweet Orange, Citrus sinensis. This made it very difficult for us to publish our own paper because a citrus genome sequence wasn't anything new any longer. Nonetheless, we persisted and we did more work, and you can see in Nature Biotechnology, an equally prestigious journal, in June of 2014, we published a paper titled Sequencing of Diverse Mandarin, Pomelo, and Orange Genomes Reveals Complex History of Admixture, and so on. So it was actually quite all right. The international group developed many new methods to analyze and compare citrus genomes, and we refuted and corrected several incorrect interpretations of the first paper that was published. We, as a group, succeeded in providing the highest quality available citrus genome sequence for the global citrus research community, and that is the Clementine sequence that we started out to produce. It is the highest quality, and that has been the gold standard until today. However, the international group continues to work, and we have some new progress that will provide new sequences that will lead us to the platinum level from the gold level. Importantly, as these cats were being herded and moving about, the relationships among the main players, the main cats, were substantially strengthened, and this led to many new collaborations, and our work continued, and a much bigger prize came to those of us who persisted, and that prize is right here. We were able in 2018 to publish in Nature. There had not been a citrus publication in Nature since the late 1950s or early 1960s, as I recall. And we published an article on the genomics of the origin and evolution of citrus. This is a landmark paper that's been widely recognized throughout the world as establishing a new way of looking at the origins and the evolution of citrus. And most of us involved were really happy that we were able to find ourselves published in Nature. If you look at the list of authors, You'll see there are several from Spain, from France, from the US, and from US, US, and US Joint Genome Institute. So a much smaller herd of cats, but we accomplished substantially greater things than we had originally set out to. This is an image that sort of covers across time as you go from top to bottom, the value and the accomplishments of genomic research and how they relate to the understanding of genome structure, understanding biology of the genomes, the biology of the disease. In this case, advancing the science of medicine and actually healing human health issues. And at the bottom, you can see beyond 2020, that's where we are today. And much of what's happened in the world of human genome sequencing and the use of that information has basically followed this pathway. And in fact, it's true that although we're behind the human genome community, the human medical community, those of us in citrus are moving rapidly forward, not only to improving and understanding the genome structure and so on, but actually applying this to real world problems such as I described at the beginning of my talk. So let's get to some take-home messages now. This is something I copied from the internet, notes on the gentle art of herding cats, but I think in some ways it's a very good summary. Things that I've learned. Cats don't like to be herded. And in fact, you can't really herd cats, though I would disagree with that to some degree. It's not so much herding, it's being a part of the herd and moving it in the right direction. A fine distinction. Cats prefer to herd themselves. Yes. Cats understand that they sometimes need to be herded. They understand it, but that doesn't make them any easier to herd, in fact. Cats don't like being reminded that they are being reherded. And harsh herding has some negative consequences. So the lessons that I've learned, and perhaps you may learn by considering some of what I've talked about today, it's important to be kind to all the cats. Take the high road. You will take some knocks for taking the high road. I certainly was knocked for taking the high road and trying to share. But do it because it's the better way to do it. However, recognize that not all cats will be kind in return to you for being kind to them. You need to have thick skin. Be like a cat and get past that. Just let some of the cats go because new cats will come along and they will find you and they will join your herd and things will improve. It's important as a leader, as a group, to define the single overarching goal. What is it that we're really trying to achieve? Where should the herd be in the end? And perhaps as a corollary to that, where can the herd go in the future? Our herd got to where we wanted to be in the end, but we actually moved much farther and continue even to this day to move forward. I would be remiss to be talking about cats and not to say something about dogs. Be doggedly determined to reach the final goal. And finally, embrace the politics. There's no way that you can accomplish the things that are accomplished, that we were able to accomplish, without getting into the politics, without dealing with it, without getting a little bit dirty. But again, taking the high road and trying to diplomatically resolve the issues, quietly, humbly, move the herd forward. So I have learned how to herd cats. It's quite simple. Look like a cat, act like a cat, hold the stick with the end goal in sight, and you'll be surprised how we all can move in the same direction. So I'd like to acknowledge at this point the folks from the Florida citrus industry, 2 organizations that were very generous in their support of this global citrus genome sequencing effort. Roche Applied Science, 454 Life Sciences, the many contributions of the US Department of Energy Joint Genome Institute, and the international funding from agencies and programs in Brazil, France, Italy, and Spain. And with that, I'll close. Thank you for your attention, and I hope you can enjoy, perhaps smell and imagine the flavor of the beautiful citrus fruit you can see in this photo. Thank you very much.
So that's the last speaker in this session, but we do have some time for questions. And I do have one question that appeared, and it was answered in the chat, but I wanted to have Carrie address this question that was asked about how does one initiate a cooperative agreement with the USDA NIFA? Could you talk about that a little bit?
Sure. So it doesn't quite work that way. You don't start a cooperative agreement and initiate it with NIFA. It goes the other way. It's an invitation from NIFA or any other grant agency, usually through what they call an RFA, or request for applications. That's the formal answer. The informal answer is you have to know what RFAs are coming out, and you can also potentially be part of the process that encourages RFAs to come out or to include projects that you are interested in. And I say that the ways to make sure that you are part of that process, and NIFA and other agencies actually do want to hear from us as scientists. What are the things that are, what's new? What solutions can we bring to the problems that government is trying to fund? So the way to get on the inside, sort of, and be part of that solution is to volunteer for grant panels. Get to know the national program leaders for various reasons and find a way to develop a relationship with them so that they know what you do, they know what you can bring to the table, and they find what you do valuable. That means that they may call you and ask for your perspective on something, or they might ask you to manage a panel or be part of a panel. Anytime you can do that, you get to understand better the inner workings of the government, as crazy as they are and inefficient. That's been the main way that I have gotten to be part of being on the development side, so that when an RFA comes out, it actually has the pieces in it that you can recognize. The other way is just to keep applying to projects and work on projects with others. Always say yes, or always find a way to say yes, so that you are known as somebody who can get stuff done. There is real truth in, if you want something done, you find a busy person. There's real truth in that, and our national program leaders use that frequently. At least I have found it to be that way.
Great, thank you, Carrie. Another question to all of the panelists is, managing people seems to be an important aspect of handling large, complex projects. Can the panelists comment on resources or trainings that they found that might be helpful for understanding how to better or effectively manage people? So either Fred or Carrie. Fred, I don't know if you want to comment on this.
Well, I really wish that there was such a resource when I began this. I didn't actually set out to be the leader. I was quite happy just to be a participant in a larger group. A former director of ours once asked me if I stepped out of the room to go to the men's room when the leader was being selected, because he recognized that this was potentially an extremely difficult and challenging job. I think, you know, it's important always to be a people person, and I don't know how you get trained to do that, but it's important to relate to individuals and across cultures. And I think I might have been selected to lead this group because I was maybe the least offensive to the largest number of people. That's not from a book, and that's not from a resource, but that seems to have been the way it worked. And in the end, I would have to say it's been successful. There are things that are continuing from our interactions. Even today, just today, we were informed that we have another paper published on a very important citrus genome, Poncirus, which is a source of all kinds of resistance genes. And the work continues. So it's something that, in my case, grew over time. There wasn't a book I could go to find out how to do this.
Okay, thank you. Carrie, can you comment?
I would say the same. I know there's probably a ton of training online through UF for us to take. That means you have to find the time to do it. And that's a frustration. I could have used it. I made mistakes along the way as I learned how to do management. We don't train people particularly well in that. IFAS does have, and UF does have some resources to help with that. The number one guiding thing for me has always been treat them like you want to be treated. Think about the things that motivate you and find a way to use those to motivate others. Be willing to step back and be the support person, not the recognized leader. So while you may be doing the leading or the herding, I always show people that my program is a pyramid flipped upside down. All stuff slides downhill. So I deal with the problems. I find the ways to keep people supported and funded and whatever so that they can do their piece. And whether that's a huge multi-state project or just here in my own program, that's the standard that I use, is make everybody else's job easy so that they can help you do your job. It's kind of generic, I suppose, but that is actually how I do it.
And that's the way it works, actually, Carrie. You're right.
Absolutely. Well, great. Well, thank you both. We're running a little bit low on time, and I don't see any other questions popping up on my screen. But I wanted to let everybody know that we will add Fred's presentation to the recording so that you can view all of those great cat slides and see the visuals that go along with everything that he shared with us today. So I appreciate everybody listening to our session today, and I think that there's a break coming up for about 10 minutes until the final session of the day begins. So again, thank you to the speakers and also to everybody that helped organize this, Cassie Sexton especially for her help in getting all this together. So thank you.
So I think we're ready to start our session. If you came on just a minute ago, you saw a large beetle from Aikido. So we are engaging our audience in science as we speak. So thank you all for joining the last session of the IFAS Research Forum. It's great to have you here, and thank you for being with us. Our session is Enhancing Research Through Engagement, and we are going to do that in a slightly different way. As you see the blue box on your screen, we would like you to go to menti.com so that you'll be able to answer some of our questions. And the code at the top, 3366755, is the code you will need to use to answer the questions. So let me introduce our panel today. We have Dr. Emilio Bruna from the Department of Wildlife Ecology and Conservation, as well as the Center for Latin American Studies. He does work in tropical ecosystems and how they're affected by deforestation and climate change. Our second speaker is Dr. Akito Kawahara, who is an associate curator at the Florida Museum of Natural History and McGuire Center for Lepidoptera and Biodiversity. He does work in insect phylogeny, predator-prey interactions, and has been featured in several videos, including PBS Sex, Lies, and Butterflies, and The Beetle Queen Conquers Tokyo. So perhaps engaging with stakeholders involves clever titles. I will be speaking. I'm Martha Monroe at the School of Forest Resources and Conservation. I do environmental education and conservation behavior. And our final speaker, who is moving us into the Mentimeter technology, is Dr. Megan Ennis, who's an assistant curator in museum education at the Florida Museum of Natural History. So thank you all for being part of this panel. And we'll go to the next slide. We're talking about engaging audiences and stakeholders of different stripes. And when we do funding, when we get funding, there's a contract, of course, where we have a sponsored program and we sign a contract for the university signs for us to do something specific. Engaging with stakeholders is a bit of a risk. They could help you change the question. They could help you change the outcome, which could mean changing the contract in a significant way. So what I'd like you to do is go to your Mentimeter question and answer this question. So you've got 4 choices. Are you willing to engage stakeholders and change the contract? Are you willing to engage the stakeholders, but only on things that won't change the contract? Would you rather not engage the stakeholders? Or would you prefer to get funding that may even encourage you to engage with stakeholders? And you're figuring how to vote. Thank you very much. This is super. I've never used Mentimeter, but it's happening, it's working. 19 people have said something. Megan, how long do we wait?
I think we'll give it a couple more seconds.
Okay.
Because it looks like everybody's being pretty heavily.
Yeah, there's definitely some diversity out there. The people who at least signed on to this forum are interested in engaging with stakeholders. Nobody's voting for that number 3rd slide. And you're recognizing that there are funding opportunities where engaging with stakeholders is a reasonable thing to do, and it's encouraged. So that's great. Some people are also willing to change the contract. What do we mean by stakeholders? There's lots of different kinds of audiences that can be engaged. There's people who care, and I think Emilio is going to be talking more about stakeholders. There's also partners who may form a slightly different relationship. Your own collaborators, the people you're working with on your research team, are other kinds of people that can be engaged. Funders can be engaged at a different level. And so we can go to the next slide. I thought it would be helpful to have a couple illustrations about this kind of engagement. Here's an illustration of where the research team can form a kind of internal engagement that could require some particular strategies that would be helpful. And we have the typical sort of researchers do something that the audience needs. However, if there's engagement with that audience, things could shift around a little bit. So the next slide suggests that if the audience is communicating back to the researcher, the whole research question may change, which would then send a different message to the audience. And the audience may be collecting data that the research enterprise will be using. Citizen science is a great example of that. And in another version, if the researchers are creating a product for the audience, and the audience is engaged with the researchers around that product, the next slide, the product may be changed. And so it may become a better product, ideally, better for the audience. So there's all sorts of ways, next slide, that we can engage with the audience. Those are just a few broad categories. If you have other ways that you've engaged with your audiences, please type them in the Q&A box. You don't have to use Mentimeter. Put them in the Q&A box. We will continue with our presentation, and we'll be able to come back to those ways and encourage more discussion about those strategies towards the end. Just like everybody else, we're aiming to get this done and save 10 minutes for discussion. So that's our goal. So for the next slide, our panelists are going to be talking about 4 different ways they have engaged with their audiences. Our first presenter is Dr. Bruna, and here you go. Thanks.
Well, hi everybody. Thanks a lot for sticking around. This is really great. It's been really informative today. And so I was asked to speak a little bit about enhancing research through engagement, and I'm going to take a kind of a slightly different approach in terms of what I talk about. Next slide, please. And I often like to start out talks in which we talk about what we do with this quote from an essay by Kofi Annan that appeared in Science, in which he writes that 95% of the new science in the world is created in the countries comprising only 1/5 of the world's population. And much of that science in the realm of health, for example, neglects the problems that afflict most of the world's people. Next slide. And this is some really interesting, nice visual documentation of that. This is by Juan Pablo Perin. It's a figure of basically the locations of the authors of all the articles that were published in the Web of Science in 2016. And you can see that that production of knowledge, of scientific information, is really concentrated in the global north, in the United States, Canada, and Europe in particular, with a couple of other countries thrown in. Next slide, please. And further went on to write that this unbalanced distribution of scientific activity generates serious problems not only for the scientific community in developing countries, but for development itself. And the emphasis here is mine. He writes, it accelerates the disparity between advanced and developing countries, creating social and economic difficulties at both national and international levels. And I think this is a really important point because it emphasizes that what we do, just the production of knowledge, the doing of the science itself, right, which is encapsulated in the research, is itself really important for socioeconomic development. It's a really important activity that we do. People who do extension and outreach and engage with stakeholders are going one step beyond that, but we really can make some important changes just by doing our science as well. And so with that, I'll move on to the next slide, which I think is another one of our questions here. We had a bit of this in our introduction, but I'd really like to ask people a little more specifically. I think we have a very kind of a particular vision of who our stakeholders are often. And so if people could go in, I think this is a poll type question, right? And so they can, people can answer, you know, more specifically, who are the kinds of stakeholders that they're engaging with? We'll give it a oh, some really interesting examples there. So producers, school and youth, community organizations, government agencies, looks like parents, growers, families, the industry, landscapers. Oh, this is great. And assuming I don't pass out from trying to lean over and try to read the one sideways here, I think that's really interesting. And I think this is a lot of the groups that we see our engagement with, that a lot of the agencies that we reach out to for funding are encouraging engagement with. So maybe that's a really good start. So maybe we'll move on here and we can flip back to this at the end in the discussion. But I'd like to emphasize that there was one group I think that wasn't on that list, right, that are really important stakeholders as well, and that's our students, right? And I think it's important to emphasize that they're are stakeholders for two reasons. They're both consumers of knowledge, which is something that we typically think of as our stakeholders, but they're also the producers of that knowledge, both you know, when they're doing their graduate study. So they play this really important bipartite role in the same way that many of the other stakeholders that we'd like to engage with do. And so this is an opportunity, next slide please, for us to think about the ways in which we can engage them as stakeholders, as well as in the capacity building function we think about. And so one example that I'd like to put forward is what we do in my class in scientific publishing. It's a workshop style class in which students are learning about not just how to write papers, but about the publishing industry and what goes into it, the kind of world of scientific publishing. Next slide. And at the very end, one of the things I'd like to emphasize here is that students have to do a research project. And this is really important because this is a class on publishing, so they're conducting research. They're learning, because of the topics of the research program, about the type of system of knowledge production in which they themselves are stakeholders. They're consumers of those papers, but they're also producing them. And it allows them an opportunity to reflect on their roles and responsibilities as those stakeholders. In other words, it's both the praxis, the practice, and also the impact of their work. Next slide. So as an example of what we've done in a couple of cases is to look at the composition of editorial boards of scientific journal to see who makes up those editorial boards. This is something we don't reflect a lot about, but it's these very small and powerful group of scientists that are making decisions about what gets into that corpus of scientific information, of knowledge. Next slide. And so as an example of the kinds of things they do, students have looked at how that composition has changed over time by selecting the kinds of journals they'd like to look at. These are typically the journals that they're interested in publishing. So you have Forest Ecology and Management, or New Phytologist, Ecology, Agronomy Journal, that kind of thing. Next slide. And some of their results. So for instance, one year, the group of students, and you can see their names there, and I'd like to acknowledge them and their contributions. One year, they reviewed the gender distribution on editorial boards to see how it had changed over the course of the last 25 or so years in environmental and natural resource management journals. Another course year, they looked at where those editors were based. So how much international representation was on those editorial boards. And it probably won't come as a surprise to people that we have found that there were nowhere near gender parity in terms of the editorial board composition, and that most of those editors are based in the United States and the United Kingdom, with a smattering from Canada and Australia thrown in as well. And so as an example of this reflection, right, they go out, they've learned to do the research, they design the experimental, the data collection with my help, they write a paper about it, that's some reflection. There's praxis built in, those papers were published. So I worked with a subset of the students then. We take the paper from class project to publication. Next slide. And there is in fact impact of these in the same way that we often think about impact in terms of our outreach with stakeholders and extension programs. For instance, one of the students was so intrigued by this, but that she followed up on this. This is Alyssa Cho, in which she looked at the gender representation of faculty and leadership at land grant and research institutions, along with her thesis advisor, and they published this paper, in one of their dominant, their most important society outlets. One of the things that I was most excited to see is an editorial that came across my desk in the journal Bioscience, in which they both cite the students' work. To this end, a recent study published by Espin et al. Serves as a clarion call for change, and in which they say at the bottom of the editorial that they're going to take up the challenge and that their future editorial boards are going to be scrutinized so they can see who exactly is a member of it. And so I think this is a really interesting example of students both engaged in the process as stakeholders, contributing to that process and making those changes. And so with that, before I step aside, I'll just say that if this is the kind of project that you might be interested in doing, you're welcome to join us. It's a crowd-sourced project in which you can download all the protocols for getting engaged. Feel free to reach out. And with that, I'm happy to take any questions afterwards about these projects and how to do this. And I'm going to hand it off to my former teammate at Manchester United, Akito, who's going to take the mic from me now. So thanks a lot, everybody.
Great, thanks, Emilio. I was not a former teammate of Manchester United, unfortunately. That would be wonderful, but yeah, I'm a scientist, and as Martha mentioned earlier, I'm at the Florida Museum of Natural History, and so today I'm going to talk a little bit about research, enhancing research through engagement from my personal experience at UF and some of the techniques and so forth that we have done in the courses that I've taught, and also in some of the lab activities that we have done. So next slide. So one of the things that we've been really focused on in my lab is, something that Emilio brought up is engaging students. And so we have a fairly large lab, and one of the internal sort of engagement activities that I've been really trying to push a lot is how to get students involved more in the process of doing science. So what we've been doing is every year we've been writing a review paper together, sort of similar to what Emilio has done in his course, in which we typically, this is pre-COVID, but we'd rent an Airbnb and go somewhere, and we would together write a paper, review paper, where everybody works on a single Google Docs, and we write different parts of it. And the goal is to get it done over the weekend or in 2 or 3 days. And this has worked multiple times, and it's extremely productive. And the students also get the opportunity to see how collaborative science can work together at the same time when we're all working on the same document together, and everybody has their own sort of responsibility for filling out their sections. And it really works very well. So I highly recommend this type of engagement, and I think the students get a lot out of it. Next slide. Another thing that we have been doing too is we've been going in these activities with the students to different places, UF facilities, RECs. We've also gone to the UF Whitney lab, where we've written grant proposals together with our colleagues. So we go as a lab, instead of just the PIs going and writing a grant or coming up with ideas, we engage the entire group together, and we thereby show the students how these ideas develop and how you write a grant proposal. I think a lot of students in general don't really understand oftentimes the kind of the details that go into big grant writing, and this, I think, exercise is very productive and helpful for them. Next slide. So I was also going to mention another way that we engaged our audience in my classes and so forth is live animals, storytelling, and time lapses and videos. And you probably, I don't know if you logged in around at the very beginning of this, but I was showing Emilio a beetle that I have right here in my office. But it's really engaging, even over Zoom. It can be incredibly engaging. Next slide. So one of the courses that I've taught in the past is a course on spiders, and my wife is actually a spider biologist, and now we teach it. We've co-teached it, and now she's teaching it mainly. But this course is about spiders, and it's an undergraduate course. And so the question is, how do we get students to like spiders more and kind of overcome their fears? And as part of this course, what we've done is we've decided to give an extra credit assignment in which students are given the opportunity to have a spider as a pet and use that pet and observe it during the course of the semester. And what they do is they observe the behavior, they write up the behavior, and that information comes back to the researcher or to us, and we help write a paper together about their behaviors. And it also benefits the students because it oftentimes overcomes their fears. They realize how amazing these kinds of animals are, and then that information gets transferred to their friends and so forth. So it's a great way to educate kids or students about natural history and observation. Next slide. Something else that we've also been doing too is part of our research in my lab is field work. So we go to the field and we go to different places in Florida, and we do a lot of sampling of insects. And through this process, one of the things that we have been doing is engaging the students and also community scientists, so the general public, to contribute photographs to iNaturalist. So many of you are probably familiar with iNaturalist, but it's an app that you use, and you can take pictures, and not only does it help you identify different kinds of plants and insects and animals and so forth, but you can also contribute your photos to iNaturalist, thereby helping understand distribution patterns and patterns of diversity and so forth that we use as scientists for our research. So this has been very helpful. Next slide. So I also wanted to mention one thing, which is that I try to engage my students in trying to make videos of everything they do. So here's just a quick video of one of our students doing a DNA extraction. And this is very simple, but these kinds of processes I think are really valuable for giving presentations and so forth to show the process. And so what we've been doing is my students film basically everything they do in the research process, so they can use them for presentations, but also in the papers that we write up, we have supplemental videos in which we not only in the paper do we explain the methods, but we also have these videos that we include as part of the methods. And this I think is great for students too, because they can really see like that we don't have to stick to the traditional sort of text of the methods, but there are new ways to show what you're doing in terms of the science. Next slide. And sort of going back to what I was saying earlier about iNaturalist, one thing we're doing here in Gainesville and in Florida throughout now is working with the Thompson Institute. So that's Florida Museum's TESSI, which Megan is part of too here. And we have this new project called the Insect Effect, and it's to document insect declines in Florida and other places as well. And this is, go next slide, please. What we're doing is we're trying to show to local to people that we need to be reducing pesticides, rewilding lawns, and using non-invasive spectrum lights and so forth to enhance the insects that are present, increase native populations of them, and also allow people to go out and document the insects and feed it back into iNaturalist and other places so that we can use that information to understand these patterns of insects and whether they're declining or not in Florida. So those are just a couple things that I wanted to mention, and I'll pass it over now to Martha.
Thank you very much, Akito. That was great. And we're zooming along at record time, so there'll be plenty of time for conversation later. Please put your questions in that Q&A box so we know what to talk about. I would like to tell a short story of engaging teachers in the process of creating some instructional materials for teachers. This was a part of Pine Map, which was a large USDA funded project to look at climate change in southern forests. Next slide. And through the process of the RFP and the proposal, we really defined what it was we were going to do. They told us we have to use our research findings to share information with with schools. We chose with teachers rather than with kids, so that teachers would share with kids. It had to be about climate and forest. So there's not much question there. So doing a needs assessment wasn't really helpful. And furthermore, waiting until we had research findings to put into the material was also equally unhelpful, because you can't wait till year 5 in a 5-year project to start developing educational materials. So we decided what we wanted to do was develop materials that would help teachers teach kids the background and the framework so they could understand the research findings, no matter what they were, which enabled us to start at the beginning. So between the RFP and the proposal, we got started developing our materials, and there were key points in time that we needed to access those teachers and other stakeholders to keep us on track. So then we asked lots of questions. Our strategies ended up being that we wanted to build capacity among these teachers and ultimately the students to understand the research findings, keeping our research goals in mind. And there were 2 kinds of research goals. There were the ones that the proposal wanted us to answer, that our forestry colleagues were focused on. And then there were our goals related to the education program that we cared about. And so we had both of those tracks going on at the same time. We used current educational research to frame our programs so that what we did would be relevant and useful to teachers. And there were 3 key ways that we engaged them prior to the publication of the final document: the advisory board, a questionnaire, and pilot testing. So I'll say a little bit more about each of those 3. Thanks, Megan, you're reading my mind. Some of that research, I just wanted to share briefly, because if any of you are launching these kinds of programs, I really encourage you to think about an educative curriculum. This means we're not just teaching students, but we're also teaching teachers. And that's what the School of Education calls it, an educative curriculum. Another useful term for us to know is pedagogical content knowledge. It's not enough to just give the teachers the information that they need to teach. We also need to help give them the activities, the techniques, and the strategies to use while they teach. And that's pedagogical content knowledge. 21st century skills is the hot button of what lots of school systems and administrators are talking about these days. We chose to look specifically at systems thinking, communication skills, and collaboration and group skills. Probably anything that comes out of IFAS could also help students develop those skills. It's not rocket science, but using that language will help administrators and teachers say, oh, I need this. This will help me. I'm supposed to be doing this. Next slide. So we started with the initial work by doing what we knew that we needed to do. We needed to take the key concepts from the research program and create some ideas that teachers could teach kids. We used a process called scaffolding, which means you don't just jump into carbon sequestration, you start with carbon cycle, carbon atom, carbon cycle, and lead up to carbon sequestration. So that helped us sort of frame out 14 different activities in this unit. Once we knew what we wanted to teach, that told us how old the kids are going to be to understand these concepts. And it also told us which classes the teachers would be teaching that could use these activities. So now we know which teachers we need to talk to. We established an advisory board that was both experts and practitioners, and we met with them every other month. And the next slide. Yes, good, the advisory board. We had 28 people. We used phone calls. We didn't have Zoom back then. So this is pre-COVID. It's hard to imagine way back then. We actually used a telephone. And we sent out these materials a week in advance with some key questions that we wanted them to focus on. We had a facilitator that was not on our team. Somebody who was very easygoing, could set the atmosphere so that they could tell us things that we might not want to hear, but we needed to hear. And we asked those advisors to review the framework, the objectives, the specific activities, and the evaluation tools that we were developing. So we worked with them for a year. Next slide. We also did this questionnaire for teachers after we got partway into the project. When we knew what the activities were going to be like, we asked the teachers, would these work for you? Would you teach these activities? And are these the objectives that would be appropriate given your curriculum and your students? This was a regional program, so it wasn't just Florida. We had to think about the national science standards and figure out what was appropriate. Since we were trying to, go back just a little bit. Since we were trying to also, unless I should be hurrying, maybe that's what you're doing. Okay. Since we were also trying to build in life cycle assessments, I wasn't really sure that a 9th or 10th grade biology teacher would want to teach about life cycle assessments. So we asked them, this is what it would look like. Would you build this into your curriculum? And this is why we think you might want to do that. And got loads of great feedback from teachers. Basically, the answer was yes, we would do that. The environmental science teachers, even more. What we got from all this was some confirmation that we were on the right track, but also some data that we could use to market the material when we finally came out with it. This is what teachers want. This is what they're going to use. This is what we're providing. It's a perfect match. Now the next slide. So we also did a giant pilot test. We paid 44 teachers to use the draft curriculum with their students. And because we were paying them, we felt like we could ask them to tell us what we needed. We asked them to do some journal entries and tell us how they used the materials and what worked and what didn't work in their classrooms. We had them use our evaluation materials with a pre-test and a post-test with students. We collected all that stuff and analyzed it. We knew that we were developing materials for high school teachers and high school students, but we had a lot of middle school teachers who wanted to use it. So we said, great, here's this. Tell us how you adapt it for younger students. And we'll put little paragraphs in each of these activities. If you have younger students, try these activities, try these adaptations. So it was perfect. They also told us where they get confused, where we needed to explain more. We found out that people who teach science in high school don't know how to help kids make a graph. So we needed to teach the teachers how to make graphs. We also got quotes like these, fabulous quotes about what they liked about our activities, which we put right into the final document. So now when teachers open up this booklet or download the materials online, they have teachers telling them how it works in their classroom, which makes it feel a little bit more accessible and, oh, I can use this too. Next slide. So I mentioned that we also had research questions. There were 4 master's students and 2 PhD students that used this project as part of their own research. So we were able to collect data that helped us answer their questions. So how do teachers feel about teaching climate change? What are they doing? What are they not doing? What are they afraid about? If we introduce carbon to students without saying it has something to do with climate change, is that better? Or if we say carbon is a big part of climate change, do they turn off or do they get more interested? Turns out they get more interested. Once we teach kids about climate change, do they get so depressed that they can't think about doing anything about it? And how do we instead teach hope and hopefulness through the process of this activity? And then how do we assess it? So research questions had to do with education, not so much about forests and climate change. That wraps up my session. And if you have more questions, you can check out that article or just email me or put them in the Q&A slot and we'll get to them later. So our last speaker for this panel is Megan Ennis, who's been our pro with Mentimeter and is going to tell us how she's used it. Thanks, Megan.
All right, good morning, everyone. As Martha mentioned, I'm Megan Ennis, and I also work in the Department of Natural History in the Florida Museum. And I'm going to talk about how do you engage the public in your research and why that's just so important to think about. we are also familiar with the calls and the need for more diversity in the scientific community, and there are a wide range of ways for us to address this issue. Because it's such a complex issue, we need lots of strategies. And one of those is starting to work with children from a young age and families to help them learn to engage in science together, because that helps to develop and support students' interests and creates a community that can help support them in pursuing those interests. So for our next question here, I want you to think about, why did you become a scientist? Who or what really inspired you, probably as a child, to become a scientist? These are all photos of me as a kid being super outdoor kid. I wanted to be a geologist and a marine biologist and all sorts of things. So for this question, we would like you to share, really what jumps out at you as one of those defining moments as a kid that made you say, or as an adult? I mean, people come to science at a whole range of ages and experience levels. So you have a little more text. Okay, wonderful. Teachers can be so important for this, and middle school is an age where we lose a lot of student interest. So I'm so glad to see that. Oh man, growing up in a tropical country, I'm sure that offered all sorts of wonderful opportunities. Oh, too many questions. We were on a talk last night from the Florida Museum called Fieldwork Fails, and someone had a quote about how, we're called researchers because we fail again and again, but we keep trying and keep trying to figure it out. That's why we're researchers, because otherwise we'd just be searchers. And I thought that was a great quote. I love all of these answers. Nature and the outdoors. Awesome. Well, being trained by a good scientist, mentors have such an important role in all of this. Parents, I love all of these. So keep thinking about those, and if you want to share some more in the chat box, we'd love to see them. But to keep everybody on time, I'm going to move along. Yes, a lack of representation is so important to be addressed. So a big part of my work is, how do we address that lack of representation in science through family-based programs? So we ran a year-long museum-based family STEM program where children in 3rd through 5th grade were invited to participate from low-wealth communities, but they were required to bring an adult with them. We let them define family however was appropriate for their family, and they came to, we offered 10 programs over the course of the year. They had really wonderful experiences. This is a picture of some of our families learning how to look for organisms in a stream, and they caught all sorts of fun things that day. And at the end of the year, we found that the students had higher levels of self-efficacy, meaning they saw themselves as being more capable of doing science. They saw themselves more as a science person, and they were engaging in more science outside of school and outside of the program. And a really important piece was, we found that our kids in the program sustained their future interest in being engaged in science, whereas our control students who were not in the program saw a decrease over the course of the year. And that is very common for this age group, which is why we picked them. But we also found that the parents saw major changes in how they perceive science, their desire to support their child's interest in science, and a better understanding of how to do that. So bringing in families and the public as a whole is a wonderful way, not just to include children in science, but also to develop a community that can better help support them as they pursue those careers. And we had 3 parents decide they wanted to go back to school to get a degree in science. So it is never too late to bring people into the fold. So knowing that the majority of us on this call today are not education researchers like I am, how do you get started in this kind of work? Well, lucky for you, there are wonderful partners on this campus and broadly that you can reach out to think about these kinds of partnerships. But I absolutely have to emphasize that you need to start this from the beginning. Good broader impacts and engagement with the public costs money. It's not free, it's not cheap. You have to pay for staff time to develop these things, as well as support for the participants to engage. So our families in these programs, we paid for meals so they didn't have to worry about when they were going to have time to get lunch. And we paid for their admission to the museum, and they even got a museum annual pass at the end of the program. So really just trying to think about how do we support our participants during this program. I just threw a couple of existing partners that are on campus that you might consider. IFAS Extension, I'm sure, has a myriad of wonderful opportunities to think about how to engage with K-12 learners all the way through lifelong learners. The Center for Pre-Collegiate Education and Training is already set up to help you translate your work into high schools and develop summer programs and things like that. The Florida Museum Exhibits and Public Programs Department will help you create research-based exhibits in the museum. If you reach out to Darcy McMahon, she can help you think about how to make that happen. And the one that I would like to plug for today is the Scientists in Every Florida Schools. So Akito mentioned the Thompson Earth Systems Institute, and the new institute received a Moonshots funding last year from the Provost's office to develop the Scientists in Every Florida Schools program. This program connects scientists to schools, particularly in underfunded, low-wealth schools across the state, and they will help you learn how to translate your research to schools. So they will help you develop lesson plans and activities. They will help you figure out how to be a role model to the classroom and just really think about how to engage students effectively. This is particularly wonderful for graduate students as they are planning their future careers as scientists, to help them think about engagement and you know, broader impacts and what that might look like for them in the future. And so if you're interested in this, do go to their website and check out all of their information. They have moved everything virtually and will help you figure out how to do this in the new post-COVID world. And I highly recommend it. All of our scientists have been wonderfully pleased with the types of projects. And this is a really great thing to put in your broader impacts, because they already have the capacity and skills to help you translate your work and have a very effective broader impacts opportunity. So do check them out and keep them in mind during your next grant proposal. So I've been watching the chat and nobody has dropped in there to share how they've been engaging with the public. This is another one of our families, and this student, they were tagging birds and learning about mist nets, and he waited all day because he wanted to release the only blue jay that we had caught that day. And as soon as he took his hand off, the bird just sat there and they had a moment and then it flew away. And so it was just really wonderful to see these students engage in ways that they had never had opportunities to do so before. So if you have any questions about broader impacts or need help identifying good partners on campus, I am happy to point you in the right directions as you are thinking about those kinds of things. And we have reached our 10 minutes of Q&A. So really excited to answer any questions for anyone on our panel about engaging our various stakeholders in our research.
Thank you, Megan, and thank you all to the panel. That was super. And does anybody have anything else they'd like to add that they didn't get to say earlier because they were going so quickly? And maybe Damien has thought of some questions.
Yeah, I just typed one into the chat, and you know, this panel I think has done such a wonderful job of providing such a thorough coverage of this topic that actually all of my prepared questions you've already covered. So I'm a little bit stumped, but maybe that's okay. Yeah, I guess the question that I do have is, what would be a good way to find out if this is the kind of thing that you want to incorporate into your research program, but without making a huge time commitment? because it seems like the sort of activities and involvements that you presented are heavy lifts and might require quite a bit of training before you'd want to maybe get in front of a group of school kids and that sort of thing. So how would you maybe dip your toe in the water a little bit and find out if this is the kind of thing that you would want to do and learn about how to fold this into your research program without making a very heavy commitment?
So
Damien, that is a wonderful. Okay, sorry, go ahead, Martha.
So I'll answer that, but I think everybody should answer that. So think about what you want to say as I say one thing. And that is that most of our departments here in IFAS have extension specialists that work with extension agents and the public, at least in the state of Florida. And it might be worth having a conversation with those people in your departments or in the counties to say, whatever it is that you're interested in, what does your research focus look like in the real world out there? What kinds of questions do people have? What kinds of problems do people have? How do people engage in the thing that you care about? Because we've found that in some of the work that we've done here, what the public needs to know, what the public wants to know, isn't exactly the same thing as what the researchers thought they were going to provide. And so that communication between the public and the researchers was really essential. It could be as simple as having a workshop before you write the proposal so that some redirection happens, or it could be that the extension specialists or agents will have some ideas about what products they can use and they can develop from your research findings that will be useful in their context. So one thing I think that represents the toe in the water is a conversation with extension people in your unit that can help expand the idea of what the audiences might be interested in. So Megan, you were going to say something.
Well, I'll just re-emphasize the idea of partners, which Martha was also just saying, because we do have a lot of different groups that are doing this kind of work already. And so you don't necessarily have to do it. Bring them on as a co-PI, talk to them, get your research questions aligned together. I mean, NSF loves to see just a good commitment to broader impacts. And so, for example, the scientists in every Florida schools, you could do it in either way. Do a one-off role model visit. They will teach you and train you on how to have an effective conversation with the one class that you're going to talk to. Or recently, one of our colleagues wrote them in to run her broader impacts part of her study. And so as they were doing research on her specific insect, she has written the scientists in every Florida school and one of their staff members into her grant budget. And they will be responsible for facilitating all of that, training her grad students on how to talk to the classes and things like that. So, scientists in every Florida school, I think, is great for wherever you are on the gamut. If you just want to dip a pinky toe in and just do a one-off, one-time class, great, they'd love to have you. Or if you're interested in including more of this in your work, they'd love to help you with that as well. But that's where my argument or my suggestion came, that you really need to bring that kind of perspective in from the beginning if that's something you want to include in your research study, because they will be able to help guide you in how to make that the most effective.
Thanks, Megan. Amelia or Akito, would you like to respond as well?
Sure. Well, I was just going to say, I wanted to just add one more thing to what Megan was saying. I think, there's a lot of opportunities on campus, of course, to do these kinds of activities. And here, I mean, at the museum, there's, we have full staff that are devoted to allowing researchers to engage with the public and so forth. So I think even if it's just a little, some idea or something not really developed, that can be pitched, and that will potentially lead into something and can help researchers, to have these different kinds of opportunities to engage with the public. I guess I was going to say a little bit about, in terms of what I was talking about earlier in terms of classes and students and so forth. I think being creative is really important, and engaging the students and asking them what they want from all this too, and developing ways in which the students can become involved more in the research that happens. And this goes back to one of Martha's initial slides with the researcher and the R and A and the arrows going back. But I think, I think we shouldn't stick to the kind of the template that we're all taught. There's ways in which we can really engage the students and have them contribute to the research while they are also involved in the whole process as well. So I think being creative and coming up with new ways to do that is important.
Thanks.
And if I could, I was going to say, I think it really depends on what this is. I mean, there are things that are increasing levels of complexity, right? We're talking about outreach. We're talking about gathering data and knowledge production from stakeholders. And then we're actually, maybe what's the most challenging thing from my point of view is engaging stakeholders so that your research questions are informed and driven by their needs, right? So that community-based or driven research is, kind of a really complicated and important thing to do. I would say that I think in addition to reaching out to people who actually do this for a living, right? We have experts on campus who specialize in this. I think a really great way to get engaged is by supporting your students who want to do it. So if you have graduate students, a lot of whom are really interested in doing this, it's very common for advisors to tell them like, oh, we don't do that touchy-feely stuff. If you want to do that on your own, focus on the science. This is science, right? Outreach is both an important component of what we do, but it's also, these things are areas of scholarship themselves. There is theory underlying it. These are disciplines that are academic disciplines where people are actively engaged in research. And so support students to do this work is a really good one. A thesis chapter that's focused on this is something that's really valuable and actually will probably help them a lot get a job down the road. The program I'm involved in, the Tropical Conservation and Development Program, actually teaches the classes where we teach students things like facilitation skills so that they can go out and do this kind of research. And it's a good opportunity for students to be surrounded by other students that are also doing community-driven and community-engaging work. So I think that's a really great way to do it, is by supporting your students and learning from your graduate students who are interested in doing it.
And I know we are out of time, but I'd like to plug, I'm teaching a class in the spring in SFRC on public communication and science communication and public outreach for graduate students. So if you have a student who's interested, please have them sign up for my class in the spring.
Wonderful. I would like to thank you all. This has been a great panel. It's been wonderful to work with you. So thanks to each of you for being here today. And I believe Damien wants to wrap up the session.
All right. Thank you very much, Martha. This was a wonderful panel and just a great discussion that we had today in our fifth and final installment of the research forum. I just want to say thank you again to all the moderators, Bob Hockmuth, Brian Pearson, and Ann Matthews, Kirsten Pels-Talinsky, and Martha Monroe, and to their wonderful panelists for this outstanding discussion today, which was enjoyed by over 100 participants. So we heard some really interesting comments that I think will stay with me. At the beginning, we heard the importance of embracing nerding out with your stakeholders and showing your passion for your research. We heard that stakeholder needs are great fuel for funding. We learned that it's important to be strategic from day one. And we also heard that it's important to find people willing to put in energy and time and be prepared to serve as cheerleader as PI of a large project. It's also important to adapt and change to stay relevant and to make space for new voices and focus on Generation Next. We learned, I think today, sort of experientially, it's important to avoid tinkering with the settings during the Zoom talk. But if you do, it's possible to remain graceful under pressure. We also heard a lot about the importance of being kind to cats without expecting them to necessarily return the favor. We also saw that Gainesville has huge beetles. Mentimeter is a really cool tool, and I think I'm going to try to use that myself. And finally, we heard that it was very important to find great partners in extension, education, and beyond, and that students are stakeholders too. And so With that, to all the faculty and staff in attendance, I just want to say thank you very much for your commitment to this important discourse with your colleagues, and I hope you have a great day. Thank you.